Good afternoon to everyone who's gathered here today for the symposium organized in honor of our very beloved Dr. Vani Santosh, who's superannuating this month. After a very long and distinguished career spanning over 29 years, 8 months, and 3 days, and during which her contributions to the field of neuropathology have been enormous. And judging by the group that is gathered here, which includes people across disciplines, including her mentors, Dr. Vasudev Rao is here, Dr. Sarla Das is online, her colleagues, Dr. Gayatri is here, Dr. Sundar is here, a lot of her friends and colleagues from various disciplines, and our own staff. It is mute testimony to her transdisciplinary outcome of her research across all of these decades. I just want to say that there is going to be a slight change in the program because our director is slightly delayed and will be joining us in an hour or so. So we will first have the scientific program followed by the felicitation. So it's my pleasure to welcome the speakers for today's symposium who have been close collaborators of Dr. Vani. We have 4 speakers who are online. We have Dr. Komori, who is a fellow neuropathologist from Japan, with whom Dr. Vani and others have been working very closely to develop an Asian-oceanian consortium for CNS tumor diagnosis. We have Dr. Kondaya, the INSA senior scientist, MRDG, Indian Institute of Science, with whom she's had probably the longest collaborations with several breakthrough discoveries that we will be hearing about. But of course, the proof of the pudding, you would say, is in the eating. So how did all of these discoveries help the patient? And that's what we will learn from the next 3 speakers, who would be Dr. Rakesh Jalali, Medical Director and Head of Radiation Oncology at Apollo Proton Cancer Center in Chennai. We have Dr. Arivarahan, our own Professor of Neurosurgery from NIMHANS, and her own colleague, Dr. Chitra Sarkar, a long-standing friend, a neuropathologist from AIIMS, Delhi, all of whom have been her very close friends in this long journey of man. These talks will also underscore how important interdisciplinary research is for patient care. And that is why the symposium is very appropriately titled Neuropathology Beyond Diagnosis, a quest to enhance patient care. I will hand over now to Dr. Nandish, who's the MC, to start the scientific program.
Very good afternoon. Thank you, madam, for that welcome note. We'll start the scientific session with our first speaker, who is from our own NIMHANS family, Dr. Arivaragan, who is the professor of neurosurgery, who has closely worked and collaborated with Vani madam. Together, they have mentored and guided many students. Sir is a surgeon clinician. As far as I know, his neuropathology knowledge is immense and is highly updated. He'll be talking on a very relevant and useful lecture, which will be useful for not just the clinicians, but also for we pathologists. That is surgeons' perspective on molecules and learning from them. Over to Dr. Arivaragan.
Slides, please. Share screen. Shall I start? Huh? Okay. Good afternoon. I thank the Department of Neuropathology and NIMHANS for giving me this opportunity. Actually, this is my duty to be here. For all the blessings I've got for the last maybe more than 15 years. So I hope I will deliver my duty promptly. So it's actually a pleasure to talk about what we have learned and what I have learned personally for more than now, say 20 years. 2003, I came here as a student. Now it's 22 getting. So let me just see the journey through my own eyes, how I learned and continue to learn from the Department of Neuropathology. And of course, the icon of the department. And today is a person who for celebration, Dr. Vani. So my interest in tumor started when I moved from my MBBS to surgery, and my guide in general surgery, Dr. Kadambari, another very successful lady surgeon, who gave me this topic, Are you work in breast cancer? So I think that is when my interest in cancer started, and this was my first paper that I published in my career on cancer. And before, I think it just got published when I joined here. And I got interested in cancer and cancer-related research from then. And it was only good when I landed here, I could continue to work on the interest. And greatly thanks to these two gentlemen. Of course, this is a recent photo, not when I was a student. So Professor Chandramouli and Dr. Anand, when I was in first year, Dr. Anand called me and said, Hari, you work in cancer. So I will send you to Dr. Vani. You go meet Dr. Vani in Department of Neuropathology. We have only listened to their lectures before and in a group, but never had a meeting with her one-on-one. And when I met, I had a very strict rules put in place. You will be given work, you have to do it. As simple as that. No excuses and no reasons to not do work. And what has to be done has to be done. And I think that's something which I follow even today. And I tell the same thing to my students. So I think in 2003, I became Dr. Vani's student, and I continue to be Dr. Vani's student till today. And I'm really happy about it. And my thanks to that will go to Dr. Chandramouli and Dr. Anand, who sent me to her in first place when I was a first year. And so I was sent to here. I intentionally retained Dr. Anita's picture, not because she started me as a mentor, but I mean, I ended up and I started being Dr. Vani's student, but then Dr. Anita remained and continued to remain as my friend, philosopher and guide, my neighbor, so many avatars, so many hats at different times. So indebted to her, we have shared our living areas, close to neighbors for many years. So there are so many personal stories. I'll not get into it. But then I learned a lot of neuropathology from these two ladies. And I'm very happy that Madam could send me this photo where they are just on both sides of me. So that I think my journey started there. And this was the first work that was given to me. These are rare tumors. This is, I'm telling, somewhere in 2003-4, before people had mobile phones and smartphones in India. And I was told to analyze 32 patients who underwent treatment in NIMHANS from 90s to 2000. The only way of communication is their physical addresses. And I had a great time roaming around Bengaluru in my old hero book, trying to find their addresses and finding out. Many times I found no buildings existed in those addresses. But nonetheless, we could get good work. And this was my first publication in neurosurgery. And that's where the journey of molecules started. And we could show that neurofilament, which was a molecule which could be identified by staining, we could see that though the tumors are diagnosed histologically, the presence or absence of a molecule can make a difference in their survival. So I completed my course, and when I became a consultant, again, these were some questions, I think, which everybody was asking. This was also in my mind. I'm going to talk about our glioma story, which is, I think, from 2006 to 2020. And I'll show the mentors who were kind enough to add me into their group. So these are 3 clinical questions. If you look at it, so I will show a lot of papers that have come in the last 15 years. Everything will look molecular. Everything will look high with loaded with technologies, cultures, and cell culture techniques, knockdown, expression, all those things. But finally, they'll boil down to these 3 questions. Why tumors of same histology or grade, like say, if 2 patients of glioblastoma are diagnosed with the same diagnosis, Do they behave differently, and do they respond same to the same kind of therapy? No. If the same kind of treatment, radiotherapy and chemotherapy, is given to two different patients, why do they respond differently? And the second question is, when we say that someone has a tumor, does he really have one tumor that you can treat that in one single way? And the third question is, whenever a tumor recurs, when we say patient with anaplastic astrocytoma or a high glioblastoma, when they say the same patient gets the tumor again after 1 or 2 years, is it the same tumor or is it different tumor? Are they biologically same or are they biologically different? Because we are doing the same kind of treatment after 2 years, thinking and assuming it's the same tumor, but it's a totally different beast. So these three questions we try to answer at different points of time in the over 15 years. And I will present this story. And my contribution should be probably a very small contribution, but I'm thankful that this great set of investigators from across, you can see people from clinical centers like NIMHANS and Satyasai, you can see investigators from Indian Institute of Science, Professor M.R.S. Rao is there, who was our team leader for so many years, and we are very happy to have worked with him. And the Dr. Sampath joined at the later half of this project. And I'm thankful that they included me as part of this project. Of course, I had to do initially some apprentice work, collecting data, recruiting patients, analyzing data, doing all that. And then I think I climbed the ropes. And whatever papers I'm going to show is the embodiment and work of so many people. These are all the research scholars who have worked in our neuro-oncology lab. And there are equal number of PhD scholars who work from Indian Institute of Science and Satyasai and JNCSR. So it's a work of a lot of people, which I am proud to stand here and present in front of you as a representative. So the work started in 2006 to 9. This was a great big grant given by CSIR for prospectively recruiting patients with glioblastoma and giving them uniform standard therapy. And there were 2 clinical centers and there were 2 basic centers. And there was also industry participation for providing the drug. All the patients received their treatment as well as radiochemotherapy, as well as follow-up imaging from the project. And we could make a very good work out of it. All these patients were regularly followed up, including radiologically, and they received standardized therapies tools protocol, what was prevalent at that time. And this was the first paper and major paper which came from that, where we could analyze why this was a prospective group of glioblastoma, when they treated equally or uniformly, why they would respond differently. So my idea at that time also was that it cannot be due to a single gene. Though glioblastoma, later we know it's very heterogeneous, it is not possible that even though how strong that MGMT is, it is not possible that a single gene can make a lot of difference for each tumors. And therefore, it was apt that with a lot of statistical analysis, Dr. Tenarazu here, who did all the statistics, that we could have come up with a group of 14 genes based on the signature, that we can differentiate groups of glioblastoma as to poor as well as better prognosis. And so we identified a set of genes, 14 genes, and you can see, and many of the genes were later taken up by research scholars to identify and explore the functional relevance in brain tumors generally and glioblastoma specifically. Some of the examples are, we worked on topoisomerase TOP2A and then identified that it actually temozolomide is a TOP2A inhibitor, and higher levels actually give better prognosis. Similar work was done on superoximate dismutase, STAT, IGF-BP3, as well as matrix metalloproteinases. As an overshoot of this work, we also did similar signatures identified from to differentiate between AA and glioblastoma, as well as that time initial work we did was on RNA profiling. Later we realized DNA methylation has got a higher relevance than RNA, and then we could identify 9 gene signatures which can differentiate between poor and good prognostic groups in glioblastoma. All these genes identified were later taken up by different students in our group to identify their functional role as well as the potential use of therapy in these tumors. I think Dr. Kondaya is going to talk about the IGFP story. And so the next question is one tumor really one tumor? And when the tumor recurs later, is it the same or is it different? So this question came up when we started addressing this question in 2010. So to give an example, if this is a large tumor and we take a sample out of here and it looks like this, then whatever study you do here, it's going to represent this. But unfortunately for us, the tumor is not like this. The tumor is like this, has different areas of different kind of gene expression, kind of invasive characters, aggression, as well as migration, as well as proliferation. So if you just sample a small area, it is not going to be representative of the whole tumor. And therefore, the biology is going to be different than what you identify in the labs. And this has been proved during this time, I think the last decade, by various studies of RNA expression, which showed that there is significant heterogeneity in gene expression profile in the same tumor when you sample it from different parts of the tumor. And so this, we had this Center of Excellence in Molecular Neuro-oncology approved for funding by DBT. This ran for 10 years in 2 phases from 2010 to 20, and we could do a lot of work based on this. And one of the, I would say, stellar work from the group was identifying various molecules which are relevant in the tumor infiltrating front. So this paper came in 2013, which actually showed if you sample from multiple areas of glioblastoma, you can find different types of glioblastoma subtypes, like whether some areas are proneural, some areas are mesenchymal. This was based on Phillips' paper, where they divided into proneural, poreneural, mesenchymal, etc. And what is actually found is a single tumor can harbor all these subtypes within itself in different parts of the area. And this paper came in 2015. We were already 5 years into this work when neuro-oncology, this was, I think, an editorial which said, do not forget the peritumoral zone. So we were working on this. And what we did is we took multiple biopsies from the peritumoral zone. When we operated this tumor, we took multiple biopsies from the surrounding areas and then identified them histologically and confirmed them histologically. This is how the tumor core would look, and this is how the peritumoral region will look, and where the percentage of tumor cells will range anywhere from 2 to 60%. We chose only those who were between 15 to 20%. And now we try to analyze, does the tumor core and the peritumoral zone, do they look similar? Do they behave similar? Do they have same kind of gene expression profiling? And as expected from us, by us, we found that there is significant difference in gene expression profiling between the core and the infiltrating front. Why is it important for the surgeon? Because when we operate, we know that we do not remove the tumor completely. And we remove the tumor as much as possible, and we leave again 5 or 10% of the tumor, either because it involves the eloquent region or because it is not identifiable during surgery because it infiltrates into the tumor. So all the studies that we do are done from the tumor core that are given to the pathologist. But all the recurrence that is happening from the tumor that is left behind in the patient, which receives the radiochemotherapy. If both are same, then the story ends there. But if both are going to be different, then what we study from the core is not immediately applicable to what is left behind. So we need to know that. And that's what this work was all about. And so we identified that the tumor infiltrating front differs from the tumor core significantly. There are a lot of genes which are overexpressed in the infiltrating front because they are the migrating front, they are the proliferating front. So they will be different from the core, which is actually necrosing because of the stagnant or the saturated proliferation. So Kritika is here. So this was her work where we identified this PDZ binding kinase was overexpressed in tumor infiltrating front compared to the normal and core. And we also identified it had a strong core, a strong aggressiveness, as well as radiochemo resistance, and contributes recurrence in tumor. And there were 4 more molecules, both Kritika and Vidya worked on it. These are all just recently published. So we are working on this tumor infiltrating front, identifying these markers, which actually make a difference in recurrence. And this would, in my mind and my hope, that would be useful for potential therapeutic targets in future. So we also did the work in recurrent glioblastoma, and again noted those markers which are overexpressed in the infiltrating front are actually overexpressed in recurrent tumor when compared to the paired primary tumor itself, which means though they are present in the primary tumor, they are present more in the infiltrating front, and therefore there is a clonal expansion of these cells which harbor these molecules, and they escape the radiochemotherapy treatment and they start growing, causing recurrence. Therefore, the treatment for primary tumor and the recurrent cannot be or may not be the same, because we cannot expect the same kind of results. And we know from our clinical experience that the treatment and the survival of the primary tumor patients is significantly higher when it's compared to recurrence, because after recurrence, whatever treatment that we give, the response as well as the prognosis is poor. And I think this could be the causes, or this could give us answers for those unanswered questions. So small note about, we also worked in anaplastic astrocytoma, initially started with various IGF-BP isoforms, and IDH we worked on when IDH was kind of discovered in this group. And when a new ISM Harlem, when the ISN Harlem guidelines came in 2016, when they said histological groups, because we had a lot of confusing names, mixed anaplastic, mixed AOAs, AAs, and oligodendroglioma, so many things, when they were all being clubbed molecularly. We had already worked on this group, and we could show that this this integrated diagnosis, molecular diagnosis, actually fares better than the WHO classification itself when it comes to predicting survival. And recently, this work is going on in anaplastic astrocytoma. Harsha is doing this work as a PhD. This was funded by ACMR. And we are working at different expression profiling molecules in anaplastic astrocytoma to identify as well as understand the behavior of these tumors. I'm not showing the results because it's her PhD work and she's yet to finish. And to conclude, it's not that we worked only in glioma. There were a lot of work which happened in medulloblastoma, where we identified the expression of IGF-BPs in medullo, and how these molecular subgroups of medulloblastoma have different expression compared to this. And we also did proteomic work and divide them based on proteomic profiling, and how this proteomic expression in medulloblastoma again differed based on the known divisions name of different pathways like Wnt and SHH pathways. And this was one of my interests 10 years ago. When MGMT was studied threadbare in glioma, we found that in fact MGMT had a role in pituitary adenoma itself, though we could not do therapeutic study. But then we found that though MGMT methylated pituitary adenomas fared better and responded better to radiotherapy. And we also did some work in ATRT. And all these works are done by a lot of scholars who worked with us, who were kind enough to be with us and carry forward our ideas. And my gratitude and acknowledgement to go to all of them. And to end, I think we had answers to some questions, but there are a lot of unanswered questions. In my mind, I think the answer will come from genes, because it is a biology that is going to drive the response to these tumors. And thank you.
Thank you, sir, for a wonderful and thoughtful lecture, running through the memory lane and journey with the molecules and learning from the molecules. I now request Vani Madam to present a small token of gratitude, a plaque to Dr. Arivalagan. Our second speaker for today is Dr. Pathuru Kondaya, who is INSA Senior Scientist, Department of Molecular Reproduction, Development and Genetics, Indian Institute of Science. Sir has been a very close collaborator and worked closely with Vani Madam in several projects, especially in the NIMHANS project and the recent DBT projects on gliomas, and has contributed extensively towards the understanding of the pathobiology of brain tumors. Madam, our students, colleagues, and friends have always spoken very highly about sir. There was a lot of exchange of students and learning between Madam's lab and his lab. Madam always mentioned about his generosity, that his lab was always open to all the students and they could carry out many works, including the basic cell culture studies. Sir has mentored and taught so many students who are in high position and doing well in different parts of the world. Madam always said that if you want to learn and observe good ethical science, you should go to Dr. Kondaya. Because of all these abilities and qualities, sir is in the befitting position as advisor to many deemed to be universities and also to the Sri Shankara Cancer Hospital. Sir has several high impact publications and patents to his credit. His one of the key research interests was in deciphering the role of insulin-like growth factor binding protein 2 in gliomas, on which he'll be talking now. And over to Dr. Kondaya.
Hello?
Sir,
Yeah, am I audible?
you're audible.
Okay. Thank you very much for the very elaborate and kind introduction. And I really don't know if I deserve all that praise what has been told in that introductory remarks. It's really a pleasure and a great honor. I'm sorry, actually, I'm not there physically. I should have been there originally planned, but for some other reason, I'm out of station. And so it's a great honor for me to speak for and on behalf of Dr. Vani. And we have been a very, very long, several years of collaboration we had in glioblastoma research. And I think she is the one who motivated us to do a lot of basic understanding of this glioblastoma. We used to have a lot of discussions. We have exchange of students, as was mentioned in the introduction. And it's really a great a pleasure to speak some work which is actually initiated by Dr. Vani, the molecule which we have worked on, at least two PhD theses have been submitted and several publications, which is called IGFBP2. Hari has mentioned, thanks to Hari, Hari, you have mentioned all the whatever collaborative research which we have initiated several years ago on Nimitli, Professor M.R.S. Rao, Professor Suma Sundaram, and myself. We were investigators from IISc. And we also have collaborators from Satyasai and also NIMHANS, of course, the primary collaborator of our research. This clinical and basic collaboration actually has taken us a notch as high, a very high profile research could be possible with these clinical collaborations. Otherwise, our basic research would not have yielded this kind of publications and the kind of research outcomes which we had in these collaborations. Now, coming back to IGFBP2, I want to share this screen. Just let me see. My screen is visible?
No, sir, not yet.
Share, okay. Is it visible?
Starting. Yes, sir. You can make it full screen.
Yeah, I've done that. Yes. Is it okay now?
Now it's perfect, sir.
Yeah. So what I want to discuss today in the next 15-20 minutes, that we have identified this IGFBP2, I think one of the groups identified. And earlier also there were some hints of IGFBP2 could be a good molecule, or a therapeutic or a diagnostic molecule in many tumors, actually, especially in glioblastoma. It's very highly expressed. And I belong to Institute of Science originally and still continue to be there, but I have recently started being an advisor in Shankara Cancer Hospital. That's what you can see my profile. So IGFBP2, we work on several things like TGF-beta pathway is one of the major goal. Several students have published research in this area. And IGFBP2 is the second most important molecule we have been working on. And several people, at least 3 PhD students have got very good results in this area. And I'm going to share some of those results. And we also worked on oral submucous fibrosis for all different clinical collaborations. Now, IGFBP2 very quickly is one of those insulin-like growth factor binding proteins, and they regulate turnover, transport, distribution, and bioavailability of IGFs. And IGF receptors are also known, IGF-1R and IGF-2R. These are all IGFs, essentially are pro-proliferative signaling pathways. And IGFBPs actually regulate these IGF. That's the primary role of IGFBPs, which was originally published. But of late, people have found that the IGF actions can be two things, two ways. One is IGF dependent, which modulate the IGF availability and prolong half-life of IGFs. And there has been a lot of literature on IGF independent actions, which shows that IGF independent action of IGFBP, some of these IGFBPs have been shown to be pro-proliferative, like cell growth and proliferation, and cell addition and migration, and apoptosis. And we show now that they are all important in tumor promotion. My data, our data, and some other data shows. With respect to cancer, IGFBP2 has been identified as one of the overexpressed gene in prostate cancer, ovarian cancer, glioblastoma, our study, and some other study, and breast cancers as well. And I show some immunostaining pictures. One of the pictures from glioma is from Professor Dr. Vani Santosh paper published in Center for, one of these epidemiology biomarkers journal. Now, breast cancer also, we have shown that IGFBP2 is overexpressed, and one student has completed a thesis on breast cancer's involvement of IGFBP2 involvement in breast cancers. And to say the least, that IGFBP2 highly expressed and upregulated GB in GBM, and the expression has been shown to be associated with increased migration and invasion of glioma cells. Many papers are there supporting us, and many people have really proposed this as a possible potential therapeutic target, actually speaking. So I will show you why we consider this as a therapeutic target in the next few slides. Now, we also have worked on breast cancer. This is a publication. I'm not going to talk about this data. And we published in Molecular Cancer that the IGFBP2 is overexpressed in breast cancers. And it's the first clue that it is actually regulating the beta-catenin comes from this study by Priyanka Sehgal. And the beta-catenin association with the IGFBP2 actions was not known before. And this is our contribution to the area of IGFBP2 research. Just to show you that the IGFBP2 regulates the beta-catenin, and this is a kind of an immunofluorescence study. We show that the IGFBP2 overexpressing cells will have a very high expression of beta-catenins. And please remember this at the end of it, when we use a inhibitory target for IGFBP2, I will show that this beta-catenin staining is abolished by using some of the antibodies which we generated. Now, we also show that the IGFBP2 regulated beta-catenin is actually one of the SRC kinase inhibitor or FAC inhibitor, we call at that point of time, PP2, and it actually can be lost at the time of when you use this PP2 inhibitor and not the other normal other things. Finally, we have done nude-mice assays where IGFBP2 overexpressing tumors are much larger. And you can see the size going up, like progressively increased tumor growth when you have an IGFBP2 expression. And this is the kind of statistics what we have. So this tells us that the IGFBP2 is an important molecule. We demonstrated that IGFBP2 expression in others, any cell type probably can, and several other cell types also we have shown this. And we can say that this IGFBP2 is overexpression leads to aggression of the tumors. And this is actually a staining pattern, which is from Dr. Vani's lab. And GBM tissue has a very high expression of IGFBP2 and also beta-catenin together. And I will show you some Kaplan-Meier curves that people who are having very high IGFBP2 expressions are poor prognostic indicators, also as well as beta-catenin. And both together actually is contributing to much more poorer prognosis. And this was a publication we made in neuro-oncology some years ago. One of the student who worked on this is Silpa Pat. And this is the publication we had at that time. It's a very important contribution saying that IGFBP2 and beta-catenin regulation in glioblastomas. And there are several other reasons why we think that there is you can see that several publications are there on insulin-like growth hormone protein driven glioma progression can be blocked clinically, blocking a clinically significant integrative kinase and NF-kappa-B network published by somebody in 2012, Kristen Holmes et al. And IGFBP2 is a candidate biomarker for one of those high-grade tumors. And so we feel that IGFBP2 it could be a very good target for one of the how we can inhibit this growth and things like that. So, what we have thought, one of the collaborators of mine, Professor Dige, in this, he's my neighbor in our institute, in our department. He works on single chain variable fragment antibodies. He's got a library. So why not we just isolate some antibodies? These are very specific. We can generate recombinant antibodies. These are single chain. It's not the regular full blown monoclonal antibodies. These are monoclonal and targeted. So we have screened this library with using IGBP2 as a protein, which we expressed in recombinant expressed IGBP2 protein, and we isolated some of the single chain variable fragments, and we showed that it inhibits glioma cell migration and invasion. It was published in Journal of Neuro-Oncology some years back. This is the data to show that the adhesion, the IGBP2 actually is one of those molecules which binds to the signaling is through integrins, and integrin binding is actually one of those adhesion properties. So we take fibronectin coated tissue dishes, and with this particular single chain antibody and fibronectin binding adhesion does not affect. And if suppose you use IGBP2 as an adhesor for the coated cells, this B7J, whatever that single chain antibody, is able to inhibit the IGBP2 binding. So it tells that the functionally this antibody can actually inhibit the IGBP2 binding to the integrins. We also show that the beta catenin is one of our target molecules of the IGBP2 via integrin signaling. I've not shown all the biochemical work what we have done, showing that it is via FAC and ILK and things like that. But sufficient to say that the beta catenin expression, which was induced by IGBP2, is abolished by using the expression. So showing that functionality of this single chain antibody what we have used. And we also show several other ways, like MMP2 RNA, which is target of beta catenin and IGBP2, is abolished. The expression is abolished by doing by treating with B7J, and similarly MMPs are also regulated, and we also show invasion is reduced, and we also show migration, invasion, and all these assays have been shown. This is a migratory property. We show that the migration is inhibited by using these antibodies. So, and subsequent to this paper, we have isolated several other monoclonal SCFs by another student, which we are now under submission. And this also shows that the several of these IGBP2 binding SCFs can actually inhibit the biological activity promoted by IGBP2. So what actually we are trying to say here is that IGBP2 is a very good target in terms of it's very highly expressed in the in GBMs, and one can actually use this as a therapeutic target to manage. So we also demonstrated that there are antibodies can be generated. Now, presently in cancer research, there are several antibody therapeutics are available. And one of the first one was trastuzumab or Herceptin, so-called Herceptin. And antibodies have proven to be very useful in many of the treatments of the cancer. Since we really list is actually becoming more and more, like we have targets for EGFR, targets for Herceptins, targets for immunotherapy, and so on. Many antibodies are available. So it is quite possible that one can generate a molecule which can inhibit IGBP2 and try it out in glioblastoma, whether this can give an approach, what we have thought that an alternate approach to treat glioblastoma. So this is where we stand, and I think this molecule has to be taken further, or inhibitors for IGBP2 should be taken further for showing its efficacy in both preclinical and clinical. That stage is still, we have not reached that stage. And I want to share some few 2 or 3 slides of very recent significant research we had with Dr. Vani's collaboration and Arivalagan's collaboration. And this idea came actually, this framework, like basis for this has been described by Dr. Arivalagan, where you're talking about differential expression of genes in the peri-tumoral region. And that led to identification of markers which probably are important for peritumoral region. And these have been published by students of Dr. Vani, and we have actually contributed molecular analysis of those cell lines and things like that. Now, means they have actually worked in our lab. Now, what triggered this, that particular finding has triggered us to look at, is there any differential methylation profile of this recurrent GBMs which compared to the primary tumor? This is a very recent work which we have done, still yet to be published. And what we have shown is that Paramita Kundu's PhD thesis is that she has already got the award and the paper is under consideration. And she found that there are hypermethylated and hypomethylated regions in the genome. And when you look at the primary tumors versus recurrent tumors, we have used about 11 primary and 13 recurrent tumors. And then we showed that there are common regulated genes between TCGA and our data sets. TCGA also has certain methylation data available. That also about 12 or 13 tumors were available. And we compared and we got with some of these lists which are common between our data and TCGA cohorts. And then what we have done is what Paramita could do is to look at the differentially regulated genes in the recurrent glioblastoma versus hypo and hypermethylation, which we mapped them on to each other and then arrived at certain list of genes which are differentially expressed in hypo or hypo. These are all validating in terms of their methylation profiles, and these are all different genes under different categories. Is just a list tip of the iceberg, and there are several genes involved in it. And she actually found that there is one gene which she was interested in called TEM8 gene, which is actually shown to be involved in some other pathways. And this TEM8 gene is actually, she actually worked out the entire pathway of, it works one through the ILK, integrins and ILK and FAC, GSK3 beta, again impinging on the beta-catenin expression, beta-catenin stabilization and transcription. So she actually showed that this particular gene, which is TEM8 we call, which is actually, she has shown increased proliferation, invasion, migration with the overexpression of this TEM8. And she also did the knockdown studies of TEM8 and then showed that there is a reversal of the aggressive behavior of some of these cells, which are glioblastoma cells. So all this suggests that this particular gene, which is TEM8, and she has other genes also in the pipeline. So this student has completed work with two characterization of functional characterization of two genes. The thesis has been approved, and then we are waiting for the publications to come out. They're all submitted. Now, to summarize my talk, what we have shown here is IGF-BP2 is overexpressed in GBM, which is not our own study, other studies also. And we showed that IGF-BP2 regulates the beta-catenin activity. This is our own finding, and others also have repeated this. And this beta-catenin activity is through integrin signaling, and which doesn't depend on IGF-1R signal. It's an IGF-1R signaling independent. And we have shown that the co-expression of IGF-BP2 and beta-catenin is indicator of poor prognosis of the patients, and IGF-BP2 promotes tumor growth in vitro and in vivo. And we also show that in principle, like, as a proof of principle, we can inhibit IGF-BP2, effectively neutralize IGF-BP2 actions, which could be a potential approach to manage the aggressive forms of GBM. So with this, I stop. And there are so many things we can talk about. One is collaboration with us, but I think it's only 15 minutes given to me. So I acknowledge the contributions are actually collaborations of, in fact, this entire NIMHANS program, which I think Ari has alluded to, and Professor Amaras Rao is the one who actually led our team, and Professor Kumar Somasundram is a very, very active collaborator of in our program. He's the team leader of our DBT, two rounds of DBT, like we had a phase 1 and phase 2 DBT, the Center of Excellence programs, where I was a co-team leader. And Rajan Dige contributed to our understanding of identifying the SCFEs. And there is no, this work could not have been possible at all without the active collaboration. In fact, I must say active collaboration, because most clinical collaboration ends up actually by providing samples, and the basic scientists work on them, and then that's the end of the collaboration. But in our case, it was very active. We had a two-way collaborations, and Dr. Vani Santosh is the one of the leading these collaborations, and she was pivotal in our research. And Dr. Ali Arivalgan is although he said his contribution is minor part, but the tumor itself is coming from Arivalgan's idea of taking different zones of tumor and things, and he very painfully did the surgical part, and that is where the beginning of our research. And I think his contribution is enormous in this. And I thank the students with Shilpa Patil. These are three principal students worked on this project. And we thank the funding from CSR, Nimitli, DBT, and thank you all for listening to me. And all the best wishes to Dr. Vani for a very comfortable post-retirement, or not retirement, but superannuation. And I'm sure she will be active, and I'm sure we will still seek her advice and collaborations in the future. Thank you very much.
Thank you, sir, for an illustrative and informative lecture and for the wonderful words.
Are there any questions we should ask? Or I think this session should not have questions. Hello, Dr. Rakesh Jalali. How are you?
No questions, sir. No questions.
Okay. Thank you very much for giving me this opportunity. It's a pleasure speaking for and on behalf of Dr. Vani. Actually, this is a very, very fruitful collaboration we had. Thank you, Dr. Vani.
Thank you, sir. Thank you. Our next guest speaker is Dr. Rakesh Jalali, who is presently the Medical Director and Head of Radiation Oncology at Apollo Proton Cancer Center in Chennai, the only clinical proton center in Southeast Asia. Sir was previously the Professor of Radiation Oncology at Tata Memorial Center, Mumbai. Sir has been instrumental in establishing the Indian Society of Neuro-oncology. Please know that in 2008, which is one of the pioneer societies in the world now, Sir was the main pillar, the founding General Secretary, President, and Senior Advisory Council of ISNO. He's a highly sought after speaker, is widely regarded for his teaching and expert guidance in various national and international conferences and meetings. Sir always recommended and supported multidisciplinary approach towards cancers and has been instrumental in guiding treatment philosophies in the management of cancers. Famed for his commitment to charity and promoting equitable cancer care for patient population in the developing world, he founded the Brain Tumor Foundation of India, an internationally recognized charity organization. His knowledge of neuropathology is amazing, and is very well versed with the WHO 2021 and all the recent update. He is so humble, friendly, and an amazing gentleman that we are all very fond of interacting with him. And Sir will be now speaking on a very useful and relevant topic for today, that is transforming neuro-oncology care in India, a brief overview. Over to Dr. Rakesh Jalali. Sir, you are muted. Yeah.
Thanks very much, Nandish, for a very kind and warm introduction. And I like to begin by expressing my gratitude to the organizing committee, Professor Anita Mahadevan, in particular, not only for asking me to participate in this select August gathering of speakers, but also sharing some of the thoughts on a subject, on a CMA, which is beautifully, thoughtfully organized on the superannuation of our dear friend Vani Santosh. So with a personal joy, delight, and a great sense of gratitude and honor, I like to share some of the thoughts on the neuro-oncology perspective, particularly in our country, what we have witnessed in the last 15, 20 years. Now, all of you know that we talk about cancer, and it is still a very difficult in general, but very rarely we talk about cancer survivors. The recent update actually shows that cancer, although still a very formidable challenge, but more than 50% are expected to live more than 10 years, particularly in childhood cancers, young adult population, and many other cancers as well. So therein brings the issue of what is called survivorship. Is it enough to just cure a cancer patient, or is it also important that that cancer patient lives absolutely normally in the society? So it has been accomplished. So this is a reasonable endpoint now in all oncology-directed therapies. It has partly been accomplished by enhanced surgical and technology, tremendous insight on molecular genetics, so on and so forth. We have already witnessed kind of a revolution after the TCGA data, especially in the last 20 years or so. Also, tremendous enthusiasm, and which was touched upon by both my previous speakers, that all the tumors, no two tumors will be similar, no tumor patients will be similar. So how to arrive at a very personalized or a kind of a personalized approach, whether it is including different targeted therapies, immunotherapies, high precision radiation, surgical skill, eventually to arrive at this fine balance of achieving long-term survival and cure vis-a-vis what is called the quality of cure or quality of survivorship. And cancer, CNS tumors in particular, unfortunately, although many of them are cured, they do end with significant challenges in the society, whether it is their cognition, endocrine, growth, education, employment, cohabiting, second cancers, many other issues which can face the patients even if they survive the tumors. Now, obviously, one of the major things, unfortunately, we still do not have a robust prospective data collection system in the country. We're largely dependent on hospital-based and regional databases. And this is just a spectrum of different papers which have been published in the last 15, 20, depending upon the different types of brain tumors that we have from the epidemiological perspective. And one of the very major papers actually came from NIMHANS group because it was one of the largest databases. And NIMHANS also not only based on the NIMHANS data, but they practically cater as a tertiary or a secondary go-to center for expert opinion. So that is very relevant for our patient landscape from epidemiological perspective. Much has been talked about glioblastoma. Suffice to say, this is just a summary which was published about 5-6 years ago, depending upon different series that we have between the East and the West. But more or less, we have established that the outcomes in general for most of the human brain tumors are consistent with what you would expect in any part of the world, at a fraction of the cost, whether it is imaging, whether it's pathology, whether it is molecular diagnostics, whether it is radiation, surgical techniques, as well as chemotherapy. For example, 70% of our patient database had temozolomide, which was 1/20th the cost of the immunotherapy. Also held true with targeted therapy, immunotherapy, and inhibitors, which are now creeping up into our clinical practice. We also realized that it is important to collaborate, and this was the effort led by Dr. Sarkar's group, but two centers collaborated together in a slightly uncommon tumor, in intracranial germ cell tumor, and we could establish that in terms of the tumor location, and there was also correlation between different parts of the world. And Dr. Komori is there with us today from Far East, which is very common, but also in the Western population, the Caucasian population. And most importantly, the outcome data was consistent with what we would expect. We also realize, however, that our country is not simple. It is not only diverse and very large in just sheer number of people who live here, but also in terms of the diversity of the cancer care. And I'm just talking about the cancer, but it can be applied for any medical care. And many, many years ago, we ran this prospective study on quality of life, but we also realized that there is social stigma attached to the brain tumors. In the Brain Tumor Foundation work that we did, so many issues were there, ranging from simple stigmatization and to exclusion of the brain tumor patients from even normal social activities, but also in childhood brain tumors, excluding them from the school activities and so on and so forth. Of course, the financial hardship was there. So we still have a tendency to protect the patient from diagnosis. Patient advocacy and empowerment in healthcare is a significant issue. Lack of dedicated neuro-oncology setup, support staff, rehab, skewed oncologists. And we have this major paradox that a small percentage can afford anything, but a vast majority cannot afford. There are social ethical dilemmas, out-of-pocket expenditure, percentage of GDP on public health is abysmally low as compared to most of the emerging industrialized nations, and how to augment compliance and follow-up. That also led us to the Indian study of neuro-oncology, and if you know, it was started many years ago now. We have a large number of members. We also were fortunate to have a very dedicated and passionate, especially in the initial formative years, And we also set up Indian guidelines because of the diversity of the care, which all of you are familiar with it. The first was on the medulloblastoma. We also now have a journal. But what some people may not realize that as much marks and as much gratitude we can show to Kumar Somasundram during his stint as our president of the ISNO, and we always invited the basic scientists to be the president. Dr. Vijay Lakshmi Ravindranath was our first president. He was very adamant and very persuasive to start a presidential award for best clinician researcher to try to inculcate the idea of basic and translational research in a hardcore clinician. And that award has been won by several people over the years, including some of the speakers that have been spoken today. But also we put the President Award in the neuro-oncology. Dr. Dev Pujari put some personal money on it, as did Dr. Somasundram. And all of you know the ISNO travel grants and travel fellowships, both within the country as well as abroad. But what Dr. Vani Santosh did outstandingly well was when the 2016 classification came. Literally within a year or two, people grappling with how to do it. We ran a series of PMEs in the National Meeting and was instrumental in guiding all of us to put so much effort to what is very well known and very well recognized throughout the world about practical consensus guidelines. And Dr. Vani would be always forever remembered for this massive contribution which has come from India, looking at practicality of the application of relatively complex and difficult molecular genetics, how to incorporate into routine clinical practice, which is easily accessible and affordable to vast majority of the patients as well as the clinicians. But also there have been strides in imaging. Now, we sometimes don't realize that imaging in India, especially with most of the MRIs as compared to the Western population, where many of the high profile imaging categories of diffuser tensor imaging, APT, RCBB, so on and so forth, complex MR spectroscopy is still in the research mode, or insurance doesn't pay, while here in a day or two, most of the MRI centers would be able to do, whether it is pre-surgery, post-radiation, post-chemotherapy, assessing between the tumor vis-a-vis the treatment response, correlating it with molecular genetics, and also now the radiomics and radiogenomics coming into such a big picture that has made a big contribution. So we are very fortunate to have outstanding imaging clinicians, imaging experts in our country to help us in our neuro-oncology care. One of the examples we spoke about glioblastoma, the corbinous childhood brain, malignant brain tumor, medulloblastoma, series of papers and series of work over the years in various centers have demonstrated very excellent and some of very innovative way to work, whether to combine chemotherapy in high risk, neurocognitive assays, and devising simple methodologies to incorporate molecular profiling. But also credit goes to AIM group and also NIMHANS group, where they were among the first ones to run off the block, literally, when the whole molecular profiling came for medulloblastoma, not one disease, 4 diseases. Now, of course, the subsites as well, how to incorporate into routine practice. And I must say that many of these innovations and time is also of essence in research and publications. So very timely, it was published to the world, and they actually set the stage for both 16 and 21 classifications, how to incorporate IHC, for example, to differentiate the several molecular subtypes in this very common pediatric brain tumor. But whatever we have said so far, and both my previous speakers have talked mainly about the research, but I want to inform all people who are attending this meeting physically as well as virtually, how NIMHANS and Dr. Santosh and all the colleagues have been the pillars of support to routine clinical spectrum, which may not be in a clinical trial or in a research methodology, for basic fundamental working and taking care of a brain tumor patient. And to go to places NIMHANS as compared to other centers because of the ease for years and years, and that is a credit to the center, that and how forthcoming and how prompt and without any cost issues, without any compromise, they would go really into that. So I just want to highlight there, I have so many cases, but just to highlight one case, 2009, cervicomedullary tumor, to be either or an astro, actually looks like an ependymoma, was treated like an ependymoma, although somebody said oligoastrocytoma, or also called an EVN, extraventricular neurocytoma. Did extremely well for 12 years after radiation. And then you have several big tumors around in the brain, the spine is still okay. What to do? Again, re-excision, came to us for further treatment, underwent operation. You can see gross total excision done again. The histologies were again very varied. Ependymomas versus astrocytoma came to us. What do? We discuss in our meeting. We find OLIG2 is positive, likely glial. IDH obviously negative. We wondered, can it be pilostic? What do? We call up, I just pick up the phone, call up Dr. Vani, always ready. She participated in our tumor board and she did the BRAF testing. Lo and behold, BRAF fusion came positive. It was a high-grade astrocytoma with pyloid features, changed the prognosis completely, and also helped us, the patient, enormously. This is just a case snippet for the thousands and thousands and thousands of the cases. All my clinician friends would bear with me how much enormous support and the confidence that we have about Dr. Vani's interpretation and her guidance is just unbelievable. Epithelioid glioblastoma increasingly recognized. Now we know that they are BRAF driven. We have targeted therapies. Also in the country, the challenge is for how long to give. There is not only the toxicity, but also the financial cost. We also recognize the G34, the diffuse hemispheric gliomas, but again, some interesting data, and we have series now. I know NIMHANS has published their series as well, but recently 3 of ours have now started the platelet-derived growth factor inhibitors, particularly dasatinib, and hopefully this will change and help these otherwise very difficult tumors. They may be slightly better than routine GBM, but ultimately the prognosis is poor. That brings us to this classic dilemma of priority is in most LMICs, lower middle income countries, including in our setup. Should we just focus all the resources and time and energy and residents and human resource to just taking care of the patients primarily, which is our primary aim and goal, but also how to put it into the research? So this dilemma is always there. It is actually much more difficult to practice in a country like ours, especially if you are from an academic institution, because we always have this patient numbers, we have so much to do, and also the infrastructure, and yet we don't have time and infrastructure and so on and so forth. So we have argued about it in various forums, but we do believe it's the key because it improves the infrastructure, reduces the abandonment. You saw the NIMHANS, NCR, IISC study, how it has helped those hundreds of patients who were accrued on this trial because you had trained human resource, better follow-up, and early access to novel technologies. And you have to find other sources. I was extremely fortunate to be associated with Brain Tumor Foundation for decades. And some of you already know the work that we have done. The growth hormone supplement, for example, we have spent 2.5 years to just help patients and children who are cured otherwise, but have growth hormones insufficiently because they cannot grow enough tall. And this has significant implications for the survivorship, how we are able to fund through raising the money from different charities. But we also must not forget the industry support, and you have to choose your industry support partners very carefully and very judiciously. They can really help you in many, many ways. There are different funding resources. All of us are aware of the sources that we go, but there are other sources also which we can access our work. I have to say that the basic and translational work, especially neuro-oncology, is kind of an example which many, many people quote. It's just a very tiny 1% of the papers that we have put up, but you already heard tremendous amount of work. And Professor Somasundaram was very kind enough to share with me some absolutely amazing work that he has just published based on potentially path-breaking. The intranasal spray, he's trying to look whether that can be put it into the brain, and that is again something very innovative and very interesting to look forward. But that brings me to the last couple of slides. The ISNO meetings that we hold annually is such a joy, and I must tell you, the 2012 ISNO, which was hosted under the stewardship of Dr. Santosh, was a path-breaking event for us, because not only we have outstanding people with Alfred Young and Ari Perry and so many other people in a milestone meeting, But we also were lucky to be given the presidentship of ASNO, and then we had the ASNO Asian Society meeting in the 2013 based on success of erstwhile ISNOs, but of course the 2012, which we always would cherish how the important meeting. So I thought I'll just give you a brief glimpse of the, which cannot be put in words, the enormous contribution, the personal friendship, the warm nature, and just incredible contribution to the growth of neuro-oncology, neuropathology. And she represents the troika, the pillars of neuropathology, which we have in our country. And of course, she is never ever going to retire, nor would we ever be able to retire. We will always pick up the phone and ask her opinion. But on behalf of all the clinicians, on behalf of ISNO, on behalf of ultimately the patients and their families who are dependent on the pathology, which is ultimately which determines the treatment, and the further, literally, pathologists are almost like gods, because they will determine whether the patient will live or not and how well they will live or not. For that, we place our immense gratitude and all the very best wishes for Dr. Vani. And again, thank you very much for including me in this beautiful symposium. Thank you very much.
Thank you, sir, for sharing your experience and also for summarizing your work and providing a lot of messages in your lecture. And I will now go to the next speaker, who is Dr. Chitra Sarkar. She is senior neuropathologist, former professor and head Department of Pathology, the prestigious All Indian Institute of Medical Sciences, New Delhi. She is the president of Neuropathology Society of India, and she was the key person and instrumental establishing the society along with other stalwarts who are there in our department, and spread the knowledge and education of neuropathology to the community, to the scientific community. Madam is well known across the globe for her immense contribution in the field, and she has many publications, patents, and guided several PhD students. And she will be now sharing her experience and Vani Madam's contributions that has impacted the field of neuropathology and neuro-oncology. Over to Madam Dr. Chitra Sarkar.
Good afternoon. Am I audible?
Yes, madam.
And if you can share my slides, please.
Yes, madam, I will share.
Have you shared, because I can't see?
You can.
Yeah. At the outset, I must express my gratitude to the organizers, especially Anita, for having given me this opportunity of speaking in this very prestigious symposium being held in honor of Dr. Vani Santosh. I have to speak on her impact on of her contributions in neuro-oncology. It is indeed a mixed feeling today to speak on and for Dr. Vani Santosh. I have had a very long association with her, which goes back to the 1990s, when I possibly first met her when she was being mentored by Professor Sarla Das in an annual conference of the Neurological Society of India, where she had come to present her paper. Since then, we have had a long journey together. She has been a very close friend, a very dear friend, and we have shared a lot of personal as well as professional matters. She has always been a phone call away, always ready to help, and always very encouraging. And that has been my personal association with her for all these years, which I cannot sum up in any words. It's a feeling, and it's a heart-to-heart connection. Coming to her professional achievements, we all know, and I repeat Dr. Jalali's statement, she has been a pillar, obviously one of the foundations pillars of neuro-oncology in the country, and therefore, and also now globally. When we enter our institutes of eminence like AIMS, NIMHANS, etc., we are told about our trinity of mission: service, education, and research. And I cannot think of anybody like Vani, who by her commitment to excellence, her hard work and dedication, her passion and unwavering determination have, at this stage of her career, achieved a total achievement of the trinity of mission in service, education, and research. Her career has been filled with exceptional achievements, and all her contributions have been exemplary. And because of her commitment, her dedication, and her passion, she have been the bedrock of all her achievements. If we go to diagnostic services, we all know she is an excellent diagnostic neuropathologist. Both nationally and internationally, she is recognized. And she has a vast, we all refer cases to her, not only from centers within India, but also from neighboring countries, with a vast expertise in diagnosing the rarest of neoplastic and non-neoplastic lesions of the central nervous system. So she has been providing all these over her career, expert neuro-oncology services to clinicians, to fellow pathologists who seek her referral and her opinion, both throughout the country and neighboring countries. But what I think is very outstanding, and which I should highlight here, is the setting up of the state-of-art molecular neuro-oncology lab. This is possibly one of its kind in the southern part of India, and it started off as a research lab, as she tells me. But what is very, very what is to be highlighted and remarkable of Vani is that she has taken what we term as bench to bedside. She has taken all this research to the diagnostic services now. And this lab is catering to providing diagnostic services and helping in patient management all over the country. This is really one of the state-of-art molecular neuro-oncology labs in our country today, which we should be proud of. Not only does it provide clinical diagnostic services, but it also has helped tremendously in capacity building for molecular pathology in the country. She holds, students come for training, she holds workshops, and numerous PhD students have been trained from this. So if I can have the next slide. The next slide, please. Can you please change the slide? The previous one, please. The previous one. This is her state-of-art molecular neuro-oncology lab. She has been kind enough to, next slide, to provide a picture of the same. And this molecular neuro-oncology lab has not only the usual immunohistochemistry, tissue microarray, it has fluorescence in situ hybridization, then you have sequencing, and it has helped in not only, as I told you, in translational research in neuro-oncology, but also now in the diagnostic services. And these are some of the panels which are being run here as per the WHO guidelines and do help the clinician in the patient management, prognostication, etc. This has supported, as I said, capacity building and numerous theses, PhDs in neuropathology, clinical neurosciences, DM neuropathologies, MCH neurosurgery. Practically everybody has used this facility as an infrastructure, and to build such an infrastructure requires a lot of dedication, a lot of hard work, and of course the knowledge to build such and maintain such a neuro-oncology lab. Next slide, please. The next, we come to her teaching and her research. We all know that she is a teacher par excellence. I think she has been a resource person in so many meetings, conferences, seminars, workshops. We all look forward to her lectures, because with all simplicity, she is able to convey the message to the point and highlight what she wants to do. She has not only trained and guided several students during her tenure, not only in neuropathology, in neuro-oncology, but she has also played a key role in starting for the first time in the country, DM neuropathology. Initially, there were only fellowship courses at NIMHANS, but now they have the only center in the country which has DM neuropathology, and several students have passed out from this institute. And it's indeed remarkable that she has been able to contribute and take a key role in starting this course in the country today. We have all heard about her research, and it will only be a repetition if I repeat what Dr. Arivardhan and Dr. Kondaya have already said of her special research interest in neuro-oncology and her focus on translational research, especially on gliomas, and with emphasis on biomarker discovery. But what I would like to highlight is that she's a team leader, she's a team player. She has never worked in silo. Most of us want to work in silo, and therefore we do not get the expected results. But she has got exceptional and remarkable achievements and success because from the very beginning, she developed this collaborative glioma research group with of NIMHANS with the Indian Institute of Science, the JNCASR, the Satya Sai Hospital, with Professor M.R.S. Rao, Dr. Kondaya, Dr. Kumar Somasundram, Dr. Hegde. The great achievements which were achieved from this glioma research group, which you have seen, was nurtured with the cooperation of many minds with a common vision. And numerous diagnostic and prognostic biomarkers were identified by this group, and Vani was a team leader in this because it was she who validated all the genes which were and the markers which were discovered by the basic researchers like Dr. Kondaya and Dr. Kumar, and she translated this by validating all these and into patient care along with Arivardhan. The prognostic and classification signatures, very early when all this was just coming into literature, they published on methylation signature in gliomas. Epigenetics was for the first time, the methylation signature, microRNA, serum biomarkers. So all this Dr. Arivardhan has touched upon, and Dr. Kondaya, but this remarkable biomarker discovery of diagnostic and prognostic markers and prognostic and classification signatures in the very early part when molecular neuro-oncology was coming in the global scene, have made an indelible mark in the history of neuro-oncology in the country and contributed globally to recognition of this group as a very active neuro-oncology research group in this country. Several patents were also filed by this group, and I must congratulate Vani and all the team members for this fantastic contribution which made a global impact in neuro-oncology. Next slide, please. The other very important contribution which Dr. Jalali has already highlighted upon, this has been a great contribution to especially in low resource countries or resource limited countries, where Dr. Vani again, as a team, she put together not only pathologists, basic scientists, clinicians, radiation oncologists, and devised this practical algorithm for approach to diagnosis of adult type diffuse gliomas based on the WHO 2016 classification, which is referred to by a lot of people, including, I'm proud to say, Professor David Louis himself has talked about it in some of his editorials, alluding to the practical algorithm being devised by Dr. Vani, and has talked about it in several scientific forums, that every resource-limited country should develop such guidelines, and this is an exemplary contribution of Dr. Vani to neuro-oncology, which is being followed in all centers, and we use this as a sort of a template for going ahead with the diagnosis, and clinicians are also very happy with this diagnostic algorithm. Next slide, please. We all know about microarrays, but the next slide. But what is very remarkable again about Dr. Vani's contribution in neuro-oncology is tissue microarray. She started tissue microarray when we were just hearing about it. And this, can we go back to the previous slide, please? The tissue microarray helped us in the biomarker discovery validation, because this is a very technology which is available in very few centers in the country. It is quite time consuming, and it needs a lot of expertise. But with her knowledge, her expertise, and her team, she was able to develop this tissue microarray technology for biomarker validation, and it is extremely useful with her. Now, can we have the next slide, please? Can we have the next slide? Yeah. Now, coming to her publications in neuro-oncology, which have really been more than 200 publications in index journals and 25 chapters in various books. But what has put India in the global map of neuro-oncology is contributions of Vani and several others in the WHO classification of the CNS tumors 2016, and now the 2021 fifth WHO fifth edition of the WHO classification, where she has contributed to this chapter on subependymal giant cell astrocytomas. So only because of the global impact of her work, she has been a regular contributor to the WHO classification, and several of her publications are also cited if we go through the reference list of the WHO. Next slide, please. We have a very close association, next slide, in editing this textbook, the Essentials of Diagnostic Surgical Neuropathology. So the first edition came after the 2016 classification, when Vani, Geeta, and I got together, and the Neurological Society of India sponsored our book to put in the essentials of the WHO 2016 classification and some non-neoplastic chapters. But we have gone ahead, and now we have just about a week back, on the 8th of December, the second edition of the book has been released, and it includes both neoplastic and non-neoplastic lesions. Anita is also now an editor along with us. And it has, Vani has contributed several chapters in this new edition of the book. And the highlight of this was that we were given just about 4 to 6 months to complete this book. And the innumerable number of discussions, phone calls, which I had with Vani and Geeta in order to complete this book within, and Anita also in order to complete this book, is phenomenal. I was sometimes very stressed that we will not be able to meet the deadline, but she always told me, Vani, in her very calm voice, no tensions, we will do it, Chitra. And this was really so soothing for me whenever I called up at all times of the day and night, that we were able to finish this project in the scheduled time and have the book released on the 8th of December 22 at the annual conference of the Neurological Society of India. Next slide, please. A few words about her contribution in the development of neuropathology and neuro-oncology in India, in terms of being the founder member of the Neuropathology Society of India. So she is a founder member. She was the treasurer, the founder treasurer of the society, and now she is the president-elect. And she has contributed tremendously to the development of the society and the educational activities of the society in terms of the webinars, the traveling school that we do, and her inputs. And the conferences which have been organized by NIMHANS, the annual conference in 17-18, and now the ASNO conference, along with Anita and other team members of the department. They have been huge successes, drawing a large number of attendees, especially to their hands-on workshops. So her remarkable role in the development of neuropathology. She has been the president of the Neuro-Oncology Society of India, during which she has contributed tremendously also to the development of neuro-oncology in India. And the capacity building, because she's a great teacher, as I have already highlighted upon. But I would like now to highlight something which many of you may not be aware. She is now the Secretary General of the Asian-oceanian Society of Neuro-Oncology, and she is playing a very central role in the next topic, which will be talked about by Dr. Takashi Komori on a new initiative which has been taken up by this society, called as the AOC Impact. We all have heard of the C Impact, which preceded the 5th edition of the WHO classification. We have now formed an Asian-oceanian C Impact initiative to develop guidelines, similar practical algorithms for the diagnosis of as per the WHO CNS5 classification in resource-limited countries. And she, being a leader in this field, is spearheading this initiative along with Dr. Takashi Komori to get all the countries in the Asian-Oceanian region together. And we have advisors like Dr. Jalali and Dr. David Louis with us, and we are trying to see whether we are able to, in the next few months, get together this initiative of putting the AOC Impact results of in the resource-limited countries of this region. Next slide, please. The next slide tells about, it is very difficult for me to sum up all her contributions and the impact it has made, but I have tried to put in a nutshell her research, her service, and her teaching, and how it has impacted not nationally, but also globally. And she has always been a great team leader. And in recognition of all her work, she has delivered several orations and received several awards, which are enlisted here. And she has been she has several patents to her credit along with her team. Next slide. She is an elected, next slide please. She is an elected fellow of the National Academy of Medical Sciences and the Indian College of Pathologists. She serves as a reviewer of many very reputed journals like Clinical Cancer Research, Child Nervous System, etc. She served as the president of the Karnataka Neuroscience Academy, president of the Indian Society of Neuro-oncology, and currently she is the president-elect of the Neuropathology Society of India and the secretary general of the Asian-oceanian Society of Neuropathology. So I think I cannot highlight all her characteristics, but 4 which have impressed all of us, and I think there is no disagreement. Team leader with creativity and vision. She has led the team with brilliance. Her strong commitment and integrity. Her skillful execution of tasks have been exemplary and an inspiration to all. Some people leave their legacy in the hearts of people, which is never destroyed. And Vani is one of them. He has scripted grace, dignity, respect, trust in the hearts of all those of us who have come in touch with her. And she has been a very helpful, encouraging, and a great colleague and friend, and a personal friend for all these years. To me, superannuation, I believe, is only an event in the journey of one's professional life. It's only the beginning of a different innings, where maybe you get more time to pursue what you have not been able to pursue in your regular professional career, travel, socialize, follow your hobbies, follow your heart. So I'm sure this is just a beginning of her second innings. She will continue to remain active, and as Dr. Jalali said, and I reiterate, she will always be there to inspire, guide us, give her advice, and I wish her all the best, a very happy, very successful, very healthy second innings. Thank you all very much.
Thank you very much, madam, for wonderfully highlighting and summarizing. And go to the last lecture, which is a special lecture by our international guest, that is Dr. Takashi Komari. Dr. Takashi Komari is the head laboratory of brain tumor pathology at the Tokyo Metropolitan Institute of Medical Sciences, and is also the director, Department of Lab Medicine and Pathology, Tokyo Metropolitan Neurological Hospital. Sir is an important contributor and resource person for the WHO and the Asian-oceanian Society of Neuropathology, active member of the Japan Neuropathology Society and other societies, including the American Association of Neuropathology. Sir will be talking on Asian-oceanian consortium to implement practical pathological assessments of CNS tumors. Over to sir.
Thank you for your kind introduction. Can I share my slide, please?
Yeah, yes, you can, sir.
Okay. Can you see the slide?
That's fine, sir. That's fine.
Okay. Good afternoon, everybody. First of all, I'd like to congratulate Dr. Vani on her happy retirement. It's a great honor and a privilege for me to be involved to her retirement symposium. When I got an email from Dr. Nita asking me to give a talk at this celebration, I asked myself, why me? And then I thought it may be because of my privilege to make a presentation together with Vani on various occasions for past years. Actually, the first time I met Vani was almost a decade ago, during the 2013 ASNO meeting in Mumbai, where Dr. Gerardi was present, we had a joint symposium with the Indian Society of Neuro-Oncology and the Japanese Society of Brain Tumor Pathology. And then Vani kindly invited me to the 2017 annual meeting of the Neuropathology Society of India at NIMHANS. And then ESMO meeting in Singapore, and then 2018 International Congress of Neuropathology in Tokyo. This Congress was also the 4th Asian Congress of Neuropathology and the start of the Asian-oceanian Society of Neuropathology, AOSNP. As all you should remember, the second Congress was held in Bengaluru last year under the leadership of Dr. Sundaram. This slide shows the structure of AOSNP, comprised of 16 countries or regions. The current president is Dr. Sarkar, the vice president is Dr. McGreen, Secretary General Dr. Santosh, and Treasurer me. Together with other councillors, we generated the steering committee of AOC Impact. Asian-oceanian consortium to implement practical pathological assessment of CNS tumors, AOC Impact, is conducted as an ad hoc business of AOSNP. As you all know, the WHO CNS-5 was published in December 2021. It moved molecular diagnosis forward, and as a result, the content overwhelmingly increased. After the publication of WHO-5, the core member of AOSNP have strongly felt that it's becoming more and more challenging to perform the recommended molecular testing in resource-limited countries. So it has been felt that some kind of localization of the diagnostic criteria is necessary to do the WHO diagnosis. The critical point is that the value of molecular tests in managing patients is not always promising at the current stage because of the lack of effective treatment. For example, in adults, chemoradiotherapy remains the standard treatment for patients with high-grade gliomas, regardless of genotypes. In children, surgery should be the first and best treatment for the patient with low-grade gliomas, again, irrespective of genetic alterations. Exceptions are when molecular target drugs are available, such as BRAF inhibitors and TLK inhibitors for NTRK fusion, but unfortunately, it's very rare. So our goal is kind of straightforward: to adopt a new WHO classification for routine diagnostic practice in a resource-limited setting, particularly in the Asian-oceanian regions. And we also believe our attempt will eventually help pathologists in other areas worldwide with similar diagnostic environments. That's why we started AOC Impact. This slide shows the structure and our members of AOC Impact. It comprises the steering committee, advisory board, and working committee. Each steering committee member is assigned to either the chair or the co-chair of one of the working groups. Next, I want to share you what we are trying to do. These are the scope of AOC Impact. And I want to stress that our purpose is to give a practical assessment to clinicians for better patient care. So our suggestion should be clinically relevant, practically applicable, and aid patient care and treatment. However, we should guarantee a certain degree of flexibility in the individual diagnostic environment. Our recommendation should be built on feasible diagnostic procedures in resource-limited settings, namely histology, immunohistochemistry, FISH, and summary sequences. The latter two would be optional. We also try to incorporate radiological information into pathological diagnosis if it is available. I also want to clarify what we do not do. We don't intend to interfere with either WHO or the C-impact body, in that we do not try to create a new entity, modify the definition of tumor types or diagnostic criteria, and not alter the general rules and applications, including NIS and NAC. This slide shows our short goals. I mean, the products we need to create during the activity. The documentation file is a sort of data set that includes all information on the target tumor type in one sheet. The second item you need to provide is a diagnostic flowchart. Of course, it might be unnecessary for some type of tumors, such as epineumomas. I think there are two types of flowcharts: molecular-oriented and histology-oriented. This slide shows a molecular-oriented one that I created from US CAP guidelines. In this chart, if it is a diffuse glioma, you apply IDH1 and histochemistry first, followed by p53 and ETRX. The alternative is a histology-oriented flowchart, which I think fits well into our daily practice. So the steering committee decided to take this type of chart for our plan. After assessment, we'll put an information into a so-called layered diagnosis format. This is an example of oligodendroglioma. Without the code deletion study, AOC impact accepted as a code-deleted oligo when IDH1 is positive and H3K23Me3 is lost. Dr. David Lewis from Harvard Medical University suggested the name histology-oriented integrated diagnosis to distinguish it from the other WHO and HRM guidelines. I think it clearly illustrates our name. In addition, this format provides a box of an impression or suggestion under the layered diagnosis box. So you could have an official WHO diagnosis at the top and AOC impact suggestion at the bottom. Another example is printing polymorphous low-grade neurotherapy tumor liang. The typical histology with heavy calcification on CT and strong CD34 immunoreactivity would support the diagnosis. In terms of radiology, actually HRM consensus guidelines recommended the use of radiological parameter for the typing and grading of brain tumors. And it said neuropathologists already use such features for classification. And I think it's true, at least in North America. The T2 FRAIR mismatch sign is not very sensitive, but a specific sign for IDH mutant astrocytomas. On T2 weighted images, these tumors are intensely homogeneous. On FRAIR, the majority become relatively hypointense in signal with a hyperintense rim. This paper is from Mayo Clinic, nicely illustrating the typical feature of code-deleted oligodendroglioma. 90% of code-deleted oligo locates in the frontal parietal lobes and shows an indistinctive border with heterogeneous enhancement. I found it helpful when the co-deletion information is unavailable. Of course, images are not always available, particularly for outside cases. So if only accessible and reliable, will include images information into pathological reports. Finally, I want to show you a kind of a representative teaching case. The imaging study revealed a hypo-intense mass in the right temporal lobe, showing a T2 flare mismatching sign. Histology with oligoastrocytoma with some mitoses. IDH1 or 132H was negative, but p53 was diffusely positive and ATRX was lost. The C-impact framework suggests this tumor as astrocytoma IDH mutant doublet shows grade 3 based on ATLX loss and T2 flare mismatch sign. However, the official diagnosis of WHO5 should be anaplastic astrocytoma NOS. This tumor turned out to be an IDH1 or 132 mutant tumor, which is rare, but this case indicated that the C-impact framework would be good enough. So in conclusion, immunohistochemistry with Sanger sequence for detecting the IDH status would provide a clinically relevant, practically applicable suggestion that sufficiently help patient care and treatment. The inclusion of clinical and radiological information is not required in the rare diagnosis, but it can be included in the comments section. Input from broad members of clinician and scientific colleagues like neuro-oncologists, neurosurgeons, and neuroradiologists involved in neuro-oncology is essential for future decision on human brain tumor classification. Finally, I want to convey my best wishes to Vani. I was so lucky working with you. You'll be missed but never forgotten. Good luck with your new journey. Thank you.
Thank you very much, sir, for sharing your experience, plans, objectives, design, and also examples for the forthcoming Asian-oceanian consortium. Now we have come to the end of part one, that is the scientific part of the symposium and today's program. And we'll be now heading towards the actual felicitation program. And I request Shivaji and team to help with the chairs. Now again, freshly welcome and wish each one of you a very good afternoon. And before we actually start the program, I would like to start with some kind of prayers for Lord Ganesha, because this is an important program for all of us. And I wish each one of you a very good afternoon. We go loss of words when we have to thank a person who has contributed so very much to the department and to the institute. We do not know how to appreciate a person who is such a great neuropathologist, both from diagnostic aspect and research aspect. And we are speechless when we have to express our sincere respect and gratitude to a teacher par excellence who has started and mentored many of the students, including me. Yes, I'm talking about the main person of today's event, the one and only Madam Professor, Senior Professor, and the Dean of Neurosciences, Dr. Vani Santosh. As a small token of gratitude, respect, and appreciation, and to honor Madam for her services, the Department of Neuropathology has organized this small symposium. For this, I request our beloved director, Dr. Pratima Murthy, and registrar, Dr. Shankar Narayano, to come to the dais and take the seats along with our Professor and aide, Dr. Anita Mahadevan, Yasha Madam, and the main person, Vani Madam. I now request and invite our HOD, Dr. Anita Mahadevan, to welcome the audience.
Very good afternoon, and it's a pleasure to welcome you all to this symposium that's being organized in honor of Dr. Vani Santosh, who I think has been a teacher, a mentor, a guide, a colleague to everyone who has gathered here. I think if any one of us is asked to talk about Dr. Vani, we'll all start furiously searching for superlatives. Come up with lots of things: a brilliant academician, a passionate teacher, an avid researcher, an astute administrator, a gracious host. I think I can go on and on, but in short, if I had to use only just one word, it would be perfect. She's been the perfect colleague, the perfect friend, the perfect wife, the perfect mother, loved by her colleagues. I think she's been the model or the quintessential woman, striking the perfect balance between professional and personal responsibilities, loved by her students. All of us here who've had the good fortune of learning from her will testify to the fact that she's a fantastic teacher. A walking encyclopedia, I would say, who can recite textbook backwards. She has a huge fan following, and many of them are here and online today, and they're all waiting to share, I think, their most treasured memories with Madam. She's also loved by the administration, I must say. She's truly a magician who can juggle any number of hats without batting an eyelid and still ask for more. She has served as the head of our department for 2 terms. She's also served as the head of clinical neurosciences, the coordinator of the Neurobiology Research Center, in charge head of transfusion medicine and clinical hematology, associate dean of neurosciences, currently dean of neurosciences. And I believe at one point of time, she was in charge head of transfusion medicine. She was dean and controller of examination. She was in charge director. She's also been in charge medical superintendent. And once she very jokingly remarked that the only position she's not had is in charge registrar. But primarily, I must say, she's a diagnostic neuropathologist at heart, an astute diagnostician who we all go running to with all our difficult cases, and her word is the final verdict. I used to very fondly tease her as a 3-cell diagnostician. I still vividly remember, I think, June 1998, when I first came in, there was this aspirate of a 3-year-old child, which I just dismissed, saying, I don't see anything. But not Madam. She found these 3 cells on which she based her diagnosis, and that's all she needed. I was shocked. I still am. Awestruck. I still am. But believe me, that patient came back to prove her right. There are several such stories of her prowess that I'm sure you'll hear throughout the evening. But it's been that very deep, intuitive instinct and her clinical acumen that has gained her her reputation as an excellent neuropathologist, not just in this country, but internationally. She has, I must say, been the steering force in setting the high standards of neuropathology in our department that is now famed across the world. She was instrumental, as you've already heard, in setting up the neuro-oncology lab that has been one of the first in the country to introduce advanced molecular tests for brain tumors, which is very, very critical for patient care. She's played also a very key role, as you just heard Dr. Komari say, in developing several practical diagnostic algorithms which are appropriate not just for our country, but also the Asia-oceanian countries as well. But her passion for that glioma cell, I think, far outweighs all else. I have a very sneaking suspicion that it replaces even her family as number one in her affections. Apologies, Dr. Santosh. She has dedicated almost 3 decades of her life to research in that glioma cell. And in the field of neuro-oncology, she has made several seminal contributions, which you just heard about, established a center of excellence in molecular neuro-oncology with interinstitutional collaborations with the Indian Institute of Science and JNCASR to focus on translational research, identified several prognostic biomarkers and potential targets for brain tumors. These efforts resulted in a huge number of publications, numbering more than 200 in several international journals with very high impact factor, as well as several patents. Some of these contributions is what you heard her contributors talk about today. She's also authored several books, including chapters in the prestigious WHO, which to us is considered a Bible. And in recognition of her contributions in the field, she has been elected as the president of the ISNO, the Karnataka Neurosciences Academy, founder member and now president-elect of the Neuropathology Society of India and Secretary General of the Asian-oceanian Society of Neuropathology. But more important than all of this, she's an impassioned teacher. And she has scores of students who are her ardent followers, mentored several PhD students, DM, MCH students. She's received several awards for the best teacher. But I think what's most telling is that almost every one of her students have won the best research paper awards for the research. She herself has been the recipient of several awards, including the prestigious Fellow of the National Academy of Science and the Indian Academy of Pathologists. And her commitment and contributions to this field, as you see, are very inspiring. And to honor these is why we organized this symposium. We're really, really thankful to our director, Dr. Pratima Murthy, who readily agreed to grace this occasion as a chief guest. On behalf of our department, madam, I request you to please accept a token of our appreciation. We're also thankful to our registrar for coming today. He has been a very close associate of Dr. Vani and a very close friend. I would request you, sir, to kindly accept a token of our appreciation. We would also like to thank all the speakers in today's symposium who have been offline as well as online. And I think they've been very close friends in her journey, and we have heard all of the immense contributions she has made in this field. And which is why this symposium was titled Beyond Neuropathology Diagnosis, a Quest to Enhance Patient Care. And I must tell you that this title was given by Dr. Arivarhan. So thank you, Dr. Ari, and thanks to all of the speakers. It also underscored how critical interdisciplinary research has been. And that is what Madam has really succeeded in. Madam, there are scores of your fans and students out there online, many of whom are just waiting to share their fond memories and reminiscences. I must say that it has been a humongous task for us to restrict the number of people who want to share their experiences and memories, just to make sure that this symposium ends at a decent hour. At one point, I think Dr. Yasha was just saying, we may have to order dinner if we allow everyone who wants to talk. So I won't take up much more time because I can go on and on. It's a special welcome to her family members. We have her husband, Dr. Santosh, who's here, and her son, Dr. Nikhil, who's online, and several other close family members who are online. It's a special welcome to them. Also, a very special thanks to our strapping young team, with Dr. Nandish, Dr. Shilpa, Dr. Rashmi, the residents and staff of our department, who put in a lot of efforts to make this program a success. And our team from both biomedical engineering the mental health education department, including Manju, who are here handling this hybrid program so seamlessly. Madam, it's a very emotional moment. We really don't want to say goodbye. I know that after Shankar sir retired, you have been the pillar of neuropathology, and your leaving creates a very huge void, which is going to be extremely difficult to fill. But we do look forward to your continued advice, support, and involvement in taking the department further. On behalf of the entire department and all of us here, we wish you a great second innings. Sure that it's going to be even more productive than the first. Thank you.
Thank you, madam, for a warm and soothing and pleasant and also emotional welcome note. And Professor Resha has closely worked with Vani madam for a long time as a close friend and a close colleague. I request Resha madam to share a few thoughts about madam.
Thank you, Dr. Nandish. As everybody has said, this is a very emotional moment, and I can't believe that the years have passed by so fast. So, but good afternoon and a very warm welcome to all of you, both on and off the dais. You have heard about Dr. Vani's from her collaborators, the amount of work that she has done, and from the HOD about all the various administrative posts that she has held, her various contributions, and truly she's been a pillar of the department. She has tread in the footsteps of the previous stalwarts of the department like Dr. Sarla Das, Dr. Shankar, and created her own blazing path. She has worn several hats as mentioned by Dr. Anita, and I won't repeat that all over again. But she's the main architect of the neuro-oncology lab, as you have all heard, establishing a state-of-the-art molecular diagnostics in neuro-oncology, introducing several techniques, and many of them for the first time: tissue microarray, FISH, PCR sequencing, tissue culture, and so on. She is the main reason why the neuropathology department has become a one-stop lab for pathology diagnosis of brain tumors, correlating both morphology and molecular diagnostics. But it wasn't always neuro-oncology for Dr. Vani. In her early career, she has worked in several other areas of neuropath, which I thought I should touch upon since it's all neuro-oncology otherwise. She has worked on neuroinfections, especially tuberculous meningitis, arthritis. All these were autopsy studies together in collaboration with Dr. Shankar. HIV, the various spectrum of pathology seen in HIV autopsies, cerebral venous thrombosis, stroke in young, role of stereotactic biopsies in neoplastic and non-neoplastic conditions. She's worked on nerve grafting in collaboration with neurosurgery, skin biopsies and neurological disorders, to name a few. Just to tell you that she wore several hats in the research front as well, before she chose to make neuro-oncology her focused research goal. But I'm here to talk a little more about the person she is and what she has meant to us in the department and to me personally. I'd like to start by sharing some lesser known factoids about Vani. Dr. Vani was born in Bangalore in an aristocratic family and is the middle child of 5 siblings. Her mother ensured that each of them learned a musical instrument and played a sport. And so Vani became an accomplished sitar player and a very accomplished lawn tennis player. I don't know how many of you know this. She has represented the state and of course her school and college in lawn tennis. And her name has appeared in newspapers too. And from personal experience, I must say, she plays a very mean game of table tennis as well. And she scalped me when we played in the in-house matches long ago in the NIMHANS Gymkhana. I first heard of Dr. Vani when I was a PG student in Mysore Medical College through a senior student. And I remember later seeing her at a conference. And I distinctly remember Vani being quite impressed and struck by you, as she was rather striking and beautiful. And that's why, a young and upcoming pediatrician was swept off his feet. And they have now been married for 40 years. So thank you, Dr. Santosh, for being here with us. I know that he has been her most strong supporter and cheerleader, and so proud of her numerous accomplishments. He teasingly refers to and introduces himself to her colleagues as Dr. Vani Santosh. So we all know by now that, of course, we all know that Dr. Vani is a very accomplished and intelligent neuropathologist with all those adjectives. I think I've run out of adjectives, but I'll try. Additionally, there are so many admirable qualities that I want to acknowledge and share some of them with you. Several adjectives come to mind, and if I were to weave them into sentences, I'd be here all day. So I'm going to just list some of them out. One is a positive attitude, ever upbeat and encouraging to all who come in contact with her. I think you've heard Dr. Chitra also say that and Dr. Anita say that. A problem solver, not a problem creator. Problem solver, adaptable, who builds consensus and takes the team with her. Focused and hardworking, we know that. Humble and committed to the department. The degree of commitment to the department and to the institute is amazing. And she's an inspiring colleague and a team player, a leader by example and a teacher par excellence. Hearing all these adjectives, you must be thinking, some of you, I mean, who are watching and maybe don't know her too well, makes her sound like a nerd. No, she has a great sense of humor with a ready laugh as it is. Sometimes making light even of a grim situation. On a personal front, I found a thoughtful and caring friend through thick and thin, both in and out of the workspace. And as life throws its challenges, she has given me much needed support and a laugh when I've needed it. Thank you, Vani. It's unbelievable how fast the years have flown and a long phase is concluding, while another beckons with new horizons and challenging goals. But with a strong common thread of neuropathology, that doesn't go away. Family, friends, and colleagues. Dr. Vani, we wish you on behalf of the department and all your well-wishers a very fulfilling, exciting, successful phase ahead with good health, good times, and lots of travel, fun, and laughter. Thank you.
Thank you, madam, for those heart-touching words. I now request our beloved director, Dr. Pratima Murthy, to address all of us and share her thoughts.
Good afternoon. I think sometimes things are preordained. Dr. Anita initially said that we were going to have a kind of formal function followed by the scientific program. But as things happened, we were able to listen to the scientific contributions of Vani and listen to the humans who spoke before. And I think that really puts things in perspective. So I think even the Ganesh Vandana would have been quite appropriate because I think Ganesha is invoked to make sure that everything goes well. But when everything has gone well, I think it's important to thank Ganesha as well. So I think I put it in order too. Dr. Vani, Dr. Peter, Dr. Rao, Dr. Yasha, all the distinguished persons in the audience, all the people who are online, Dr. Santosh, the rest of your family members who are online. I think this is a very momentous occasion. We listened to a lot of accomplishments of Vani and her contribution, her immense contribution to the Institute, both academically as well as administratively. We've had several adjectives spoken about Vani, but I don't know how many of you know Vani as long as I have known her. So I was a wide-eyed entrant into Bangalore Medical College in 1979. And there are two seniors whom I recall with great affection. One, of course, was Anisa Hasan, and the other was Vani. So, although we know we didn't interact too much at that time, nor have we professionally interacted too much at NIMHANS either, both Anisia and Vani have been greatly inspirational figures, mostly I think because they are so caring and compassionate.
and they're very different in, in many ways. But I think that's what really defines them. There were so many adjectives that were described and attributed to Vani to describe her. I must add that I had thought about a few myself. And there actually are some additional ones to both what Anita and you have said, Yasha. And that I think stands for, I just thought I'll only talk about a few adjectives because I think most of the others have already been talked about. And that was what Vani stands for to me. Actually, Vani is a very vivacious person. And you mentioned her being very, I think Yasha talked about various aspects of her physical attributes. I think her personality really rounds it up. And she's a very, very well-rounded, a 360-degree personality, if you will. Obviously, you've heard what a visionary she's been within the area of neuropathology, how much she has achieved. And I'm very glad to hear that, of course, in addition to neuropathology, neuro-oncology, the fact that she's worked in so many different areas and has thought through and is now one of the leading lights in the area. And that has been talked about in terms of the administrative positions that she has on various associations, the kind of contribution she's made, the patent she's got, and so on. Vani is also extremely versatile, and her ability to adapt to difficult situations, I think both professional as well as personal, stands her quite apart from many others as well. There are lots of adjectives in terms of the A for the A in the Vani, and that's been spoken about. You described as an astute clinician, which of course she is. She's also extremely approachable. And I think that has been very obvious when she has been dean, that, she's been able to listen to students, sort out their problems, try and look at easy solutions rather than complicating already complex situations. She's extremely accommodating. As Anita mentioned, she would not say no. She would look to solve problems rather than create problems. And that, I think, is a great, great kind of temperament that you see is very, very useful, particularly in administrative roles. Vani is extremely affectionate. Vani, I must recall on this occasion a time when there was this business. I still don't understand many of these things, but there was this business of some kind of percentage and promotions. I didn't even know about it till you called me and actually consoled me and said it's okay. So I recall the great affection that you kind of expressed in terms of calling. Similarly, I know how, how kind of a personal cheerleader you have been for me. And I really appreciate that. You're extremely nurturing. And again, we heard how nurturing you are in your various roles. We, of course, haven't seen the personal front so much, but we've seen your ability to nurture people within your department, whether they are colleagues, whether they are students, whether they are peers. I think that's a wonderful kind of quality that all of us admire immensely. The I, of course, I don't have to talk about your intellectual abilities. I think a lot has been said about that, and that's been kind of very well recognized nationally as well as internationally. You certainly have been an inspiration to the two young ladies sitting on either side of you. Not so young, but still younger. And I'm sure you continue to be that for many, many people, not just young ladies, but also the young men and others in your department. Of course, the amount of interest you take in other people, I think that's, again, a very valuable kind of quality that you've told us. And several people mentioned the integrity factor, which I think is also very important. And of course, there is always the full S, the Santosh, your bubbly laughter, I think that keeps us all kind of, happy and, wanting to do more work. I also was very touched that there are several people online, both colleagues of yours in the past, colleagues who continue to work with you. I also saw Dr. Kanishka's name mentioned. So I hope Dr. Saraladas is also listening online, because again, they've been very important mentors to you. Dr. Saraladas has been an important mentor to you, and I'm sure will take great pride in the work that you've done. So also your several predecessors who will also take the pride. I know that you have worked very, very closely with many departments, particularly the Department of Neurosurgery. So it's nice to see Dr. Chandramouli here. and several other distinguished scientists with whom you worked, and current colleagues also in neurosurgery. But I also see colleagues outside of neurosurgery in several other departments. And I think that speaks of, the kind of respect that we have for you. As was Dr. Chitra, I think, mentioned, I mean, this is just a journey, and all of us get on the train and get off the train at different points of time. But I'm sure you will have many more journeys, many more destinations. And I'm sure that whatever you do, you'll do it very well. I hope you continue to support the department, in many ways. I don't think, you need to feel so, so kind of feel such a void, because I'm sure the contributions that people make in these departments will, outlast their physical presence in the department. And I'm sure that will be a great strength for you to, to take the department forward. And I'm sure Vani is not going anywhere. In mind and in intellect and in spirit, she will continue to be with us and support the, functioning of the department. So congratulations, Vani, on a very successful career, and congratulations on a well-deserved superannuation, and all the very best for the future, both to you and your family. Thank you very much.
Thank you, madam, for those warm and encouraging words. Now, may I request our registrar, Dr. Shankar Narayan Rao, to say a few words.
Very good afternoon, and it's a momentous occasion to recall the contributions of Dr. Vani. Our director, Dr. Pratima Murthy, Dr. M.R.S. Rao, Dr. Chandramouli, Dr. Vasudev Rao, and many of the senior colleagues who was part of the journey of Dr. Vani. They're participating in online. So I think, the day when I joined NIMHANS, last 33 years, I know her. So what is most important thing is that I think, the kind of care which she has for not only to the colleagues, but most importantly for the students. In fact, she has gifted several students to me to ensure that, okay, the PhD programs are completed. Even after their completion of their degrees, she does the follow-up. That's very, very unique, actually. As an excellent pathologist, and the way she established the molecular pathology specifically to the neuro-oncology, and it was built brick by brick, even from the foundation of the Neurobiology Research Center, and we are part of it. But our contributions are, is immense, and the passion to her field, and also more importantly, the kind and affection which she has exhibited to everybody, and her leadership. But she has one unfinished work yet. Currently, she is the team leader for establishing the central laboratory complex. And she promised that, okay, she is going to, even after superannuation, her mentorship and guidance to establish the new facility. It's a huge facility. Under one single umbrella, you have an excellent diagnostic services. On behalf of NIMHANS, I request her to spare some time, and I am sure that she will have little more time than what she is have for the laboratory complex under mentorship. I'm sure that, okay, her administrative skills, more importantly, she is an excellent listener. First she will listen everything, and she will say that she will take some break. After some time, she will tell that, maybe can we discuss tomorrow, Shankar? That means today she will clearly examine what will be the decision tomorrow. And next day she will say that even the complex issues, and she will make it very simple, and she arrives the solutions. That's the greatness of Vani Madam. And I'm sure that the kind of support and the mentorship will continue beyond your superannuation. It's only a break. But you have more time to the family. And thanks to Dr. Santosh, sparing her for long hours of all the diagnostic services, several meetings, and meetings will end very late. And the kind of family support and the support was received from the entire department and from our colleagues. Because of that, she could hold several administrative positions. So NIMHANS is ever grateful to your contribution, madam. And we are looking forward your mentorship for the coming years as well. So thank you very much. Dhanyavad, namaskar.
Thank you, sir. Now it's the time to felicitate Vani Madam. I request our director and registrar to conduct the felicitation. And as we prepare for this felicitation, I want to bring in some philosophical thoughts. As the name Vani is so apt to Madam, it symbolizes Lord Goddess Saraswati or Sharadambe, the goddess of knowledge, goddess of wisdom. Now, leaving away the philosophy, coming to just literary, it is Vani means eloquent of words. So just thinking, somewhere I heard in one of the Sanskrit verse that for success or elevation in life, you need some aspects of Vani. That is Amrita Vani, that is with those words which are very good. Divya Vani, which has divinity. Viveka Vani, words of wisdom. And of course, in the present world, Madhura Vani, which should be sweet. I think Madam encompasses all those things. You can see that our words are covering every aspect of this: sweet, knowledge, wisdom, and guidance. And it has gone to such a that I even found Vanipedia also. In the sense, it's not just Wikipedia, Vanipedia. Of course, that is more philosophical in the sense which has filled in some essence of Vedic knowledge to preach and teach the science of consciousness in the world. Of course, related to ISKCON. No, just that was a new thing which I wanted. This was just, I was telling just to make some, as there is time for preparation. Now I request our director and registrar and the dignitaries to conduct the felicitation. Thank you so much, Madam and sir. Also, Dr. Kumarvelu Somsundaram, one of the close collaborator of Vani Madam, he could not. Dr. Kumarvelu Somasundaram could not come and attend this program, so he sent a small token of gift for Vani Madam, and I request Yasha Madam to help with this.
One photograph, I think, was that's one, sir.
Please. Yes.
Please.
I thank the I thank the director, registrar, and other dignitaries to take the seats in the hall. Now, it's my honor to invite the one and only, the most important person of the event, the day, that is Vani Madam, to give her much awaited talk for which we are eagerly waiting and keen to listen. Over to Vani Madam to share her journey and experience in neuropathology. Please.
Where are the slides? How can I go to the slide? Escape. Mine is here. This one. Yeah. Good afternoon to everybody. I'm so overwhelmed with what I've heard so far that I don't even know what I'm going to speak. I mean, I don't have any words. I don't have words to thank you all. I don't have enough words to show my gratitude to everybody with whom I have collaborated, with whom I have worked. What I thought was, I won't make it too long. Don't worry, I won't hold you too long. But I thought that I will speak very briefly about my journey in neuropathology, which has been an invaluable experience for me. First of all, I should say that I'm humbled and thankful that so many of you have considered my retirement symposium worthy of your time, and you're here with me here today and also online. I'm basically here to express my gratitude to this great institute and also to all of you people. And it's been a long journey together. And during this journey, I've been able to build myself. George Burns, he's a late actor and comedian. Once he said retirement at 65 is ridiculous. When I was 65 years old, I still had pimples. And I do agree with him on that. I still feel energetic. I feel young. I can't even believe that I'm going to superannuate. So I have those sort of feelings inside me. But joking aside, standing here before you today makes me recall how 30 odd years ago I decided to take this journey that brought me to NIMHANS and to start my career as a neuropathologist. Mine is a kind of a fragmented memoir, it's a sort of illustration that is broken into fragments, and some of them even seem to be a little bit unconnected, but I'll try to put that together. If I go back to the very beginning, I mean, when I finished my MD in pathology, I was already married by then. I was in a dilemma as to which field I should further specialize in. So my husband, Dr. Santosh Kumar, who is here with us today, at that point of time told me, why don't you explore neuropathology? Why don't you explore genetics in tumors, is what he said. I was fascinated with neuropathology because neuropathology is that specialized discipline which has a broad application across clinical and research fields. As a clinical service, it defines diseases of the nervous system, either through the macroscopic, microscopic, and even ultrastructural examination of biopsy and autopsy material. And as a subject, although it is deeply rooted in general pathology, it has seminal links with both basic and clinical neuroscience. So it's such a complex subject. It is so clinically oriented, and given its complexity, it has developed in a different way when compared to other branches of pathology, and that is reflected in the practice of the subject. And that is what enticed me to come into neuropathology. So I chose to specialize in neuropathology, joined this department as a senior resident in the year 1989. I'm very thankful, very grateful to my teachers and my mentors, Professor Sarla Das, late Professor S.K. Shankar,
They have played key roles in shaping my career as a neuropathologist. They've supported me throughout my tenure in the Department of Neuropathology. Not only have they been fantastic mentors to me, but they have also taught me how to mentor other people. And I will remain forever indebted to these great role models. I had other teachers and mentors. Dr. Vasudev Rao, he's here today with us. He was associate professor when I joined the department as a senior resident. And he taught me the nuances of surgical diagnostic neuropathology, which was a great learning for me. Dr. Asha was the assistant professor at that time, and she did a lot of hand-holding because I didn't know anything in neuropathology. We learned ultrastructural pathology from Dr. Ram Mohan, and I learned a lot of blood banking technique at that point of time from Dr. Saubhagya, who was there in the department because it was a single department for a very long time. So as a senior resident, I had all these assignments, grossing of surgical specimens, assisting in surgical biopsy reporting, and then conducting clinical autopsies. That was a big job that was given to the residents, assisting in the brain cuttings, entering reports, and carrying out work in the blood bank or clinical pathology, many other work that all the residents are now very familiar with, as far as the jobs go. But I'd like to share only a few of my experiences as a senior resident. So this was my first autopsy in the year 1989, when I just joined the department. And as soon as I joined the department, there was a call from the mortuary saying that there is a case for postmortem. And I was sent there to perform the autopsy. And I was so nervous at that point of time because as trainees in MD pathology in Bangalore Medical College, we didn't have that hands-on workup sort of experience in the conducting autopsies of removing the organs. But when I went there to the mortuary, I found this very pleasant person, Mr. Mani. He's Mr. Vishwambaram. We fondly call him Mani. For so many years, he's been with us. A mortuary technician, so proficient in his knowledge and such a great experience in performing autopsies, removing the organs. And he gently taught me how to remove the brain. This is a 16-year-old boy who had succumbed to intracerebral hemorrhage. And when I took out the brain and saw the brain, I saw all these huge blood vessels that were there at the base of the brain that were all thrombosed, and I had no idea what it was. So what I did was, I said, better I don't do anything about this. I'll put it in a bucket, fill it with formalin, bring it to the department. I showed it to Dr. Shankar and to Dr. Sarla Das, and they both had a look at it and said, Vani, do you know that even we don't know what it is? It's a very rare case and we are not sure what it is. That made me feel very relieved at that point of time. Later, we diagnosed this as a dolichoectasia, and this was has been mounted in the museum for a very long time until recently. Dr. Anita took so much interest in this particular case. She removed the brain from the jar. She took sections from the blood vessels. You can see that area where she's taken sections from. And she saw that there was dense vasculitis over there, and then went ahead to do molecular testing with her collaborators at JNC, and then showed that this was a herpes zoster vasculitis associated with HIV. And that was a huge learning for me from the autopsies. Later, autopsy became a routine because we used to have about 250 to 300 clinical autopsies during those days every year. I went routinely to perform autopsies, and I've learned a lot from gross morphology. And this is what Dr. Shankar taught us all. And cases of stroke, ischemic infarct, aneurysms, hemorrhagic stroke, or even infections, as you can see here. And as Dr. Yasha said, I did have interest in neuroinfections when I came in, because I was so influenced by what Dr. Shankar was doing. He was such a role model. I just followed his path. I was shadowing him for so long. So I got interested in neuroinfections and started work on tuberculous meningitis. At that point of time, we analyzed more than 200 brains. This continued when I even became a faculty. And we were looking at the involvement of hypothalamus and even the patterns of vasculitis in tuberculous meningitis. And later, I went on to study the opportunistic infections in HIV and several other infective conditions. I also had the opportunity to analyze the brains of patients who had succumbed to cerebral venous thrombosis under the mentorship of Professor Sarla Das. So the next job was to assist in biopsy reporting. And during the early days, the biopsy reporting was based on very simple techniques. It was just H&E and special stains. Special stains are the neuropathology special stains. They need to be patented from our department, if you ask me. And when anyone here says special stains in neuropathology, the person that will flash in our mind is Mr. Ramkumar. And Mr. Ramkumar is there today with us. He was the technician who was an expert in special stains. And he was ably assisted by all the other technicians, Mary, Jagannath, and so many others. And we were able to give some of the diagnosis using only such, basic techniques. Now the department has grown quite enormously, and I have borrowed this slide from Dr. Anita. She made a wonderful collage of the different sections of the department where we have many more skilled technicians, we have project assistants, we have students who are working, lot of advanced technology to provide the state-of-the-art diagnosis as of now. But during residency, it was not just work and autopsy and reporting. We had fun times. And Madam Sarladas always, made us feel that we were a part of the neuropathology family. And we had fun times in the department, outside the department. And this is one such picnic that we went long back when I was a resident to Bannerghatta National Park. And you can see Dr. Ram Mohan's wife over here, his daughter, and also my son, Nikhil, who is enjoying. He was very small that time, enjoying that picnic. Then I joined faculty, and like any other faculty, I was involved in diagnostic services, in teaching, and in research. And I think you've heard a lot about it from the earlier speakers. But I've been really passionate about diagnostic neuropathology, and that has kept me going for many, many years. I never feel it's a Monday morning and I have to go to work, because I'll always think that there is something challenging, something that I need to go, my passion is there. And during those days, we had this consensus reporting format, and all of us used to see all the slides and discuss the cases. And the reporting officer would take the most appropriate diagnosis and pen that down. Many clinicians used to join us for reporting. You can see Dr. Indra sitting there with us for the reporting session. And we used to enjoy with biscuits and snacks and other things. The reporting was a lot of fun during the early days. But I've learned invaluable lessons in diagnostic neuropathology. Because when we look at biopsies on a day-to-day basis, we see so many cases. Many of them are routine run-of-the-mill cases, but there are biopsies that don't show typical features. And they don't show the typical textbook features, And if you look at textbook, if you look at the statements that they make in textbooks also, it says that 70% of patients with a particular disorder will have histopathological features A, which implies that 30% don't have those features. And we need to recognize that 30%, which are the atypical cases or the outliers. And that is what I have learned. And that has been taught to us by teachers and mentors like Dr. Shankar and Dr. Sarladas, Dr. Vasudev Rao, who taught us to recognize those atypical cases. And I think I became more and more confident as a diagnostic pathologist only because of that. And now it is the present era. There are though we moved from light microscopy to the electron microscopy, enzyme histochemistry, and others, present era, you can see that it is flooded with the molecular techniques, all types of molecular techniques, and the opportunities have never been greater. And I always tell my younger colleagues and my students also that you need to be at the right place at the right time. You're all here at the right place in the right time. Everything is available to you for providing a state-of-the-art diagnosis, but we learned it the very hard way. Now, teaching is the other thing that I've been very interested in and passionate about for several years. And I have seen a lot of changes in the form of teaching over the years that I've been here. I mean, I've seen the evolution of lecture materials go from chalk and blackboard, it goes to the whiteboards, the transparencies, the Kodachrome slides that we used to use on carousels, and now eventually PowerPoint presentations and videos also. And students, they used to remain very silent, obedient, compliant to the rules during those days. But now they're not like that. I mean, I'm sure my students know that. They're more empowered, they're outspoken, they're confident. I'll tell you, they're confident. And sometimes they can be even demanding. And as teachers, we spend so much time with our students during the course of the year. And many times we share life lessons with our students. And I think that has a far greater impact on the students than teaching any standard-based content. And I've had the privilege to teach neuropathology to several students. It's just not students of our department, the PDF or the DMs or the PhD scholars, but other departments, MCH, neurosurgery, neurology, psychiatry, biostatistics, neuroradiology, taught students from several other disciplines and from students who came from outside and got posted into the department. And I've enjoyed all that. And let me tell you that we as teachers learn a lot from students. So the more we teach, the more we learn. That is what I have, is my personal experience. But it's not always teaching and learning and diagnosis and work. We've had a lot of fun times and we continue to have these fun times in the department, which is such a nice break, you know. Now, may it be a birthday party or a send-off or even a puja. We've enjoyed all those events. So you've heard a lot about my research. I just won't go into the details of my research. I have been passionate about my research, though I started off with several other things like infections, and then I did stereotactic biopsies and several other aspects of neuropathology. My passion was in brain tumor research and carried out translational research. And what I believe, which has already been said, is that research has to be a multidisciplinary kind of a program. Otherwise, it's very difficult to conduct research and you will not get results also. So I've had the pleasure to collaborate with the departments of neurosurgery, clinical neurosciences, radiology, biostatistics, and even my collaborators from outside other institutes to conduct all these research. And you've seen this slide from Dr. Ari earlier, and this is our in the past 2.5 decades, you know, all my research was diverted towards biomarker discovery in glioblastoma. And we had a fantastic collaboration and with Jawaharlal Nehru Center for Advanced Scientific Research, Indian Institute of Science, Satya Sai Institute of Higher Medical Sciences. I'm very grateful to Professor M.R.S. Rao. He's here today with us. He's been our team leader, our mentor. He taught us how to perform research, how to put up research proposals, how to carry out research, how to analyze the results. It was wonderful to learn from him. My very good collaborators from Indian Institute of Science, Professor Kondaya and Professor Kumaravel Somasundaram, they are excellent molecular biologists and they taught us so much about pathobiology of these brain tumors. Professor Kumaravel Somasundaram has also been our team leader in the DBT projects when we got the Center of Excellence in Molecular Neuro-Oncology. But I want to talk about my clinical colleagues, Dr. Hegde, first of all. Dr. Hegde and Sridevi Hegde, his wife, were the ones who first inducted me into this collaborative project. Actually, they had started off the project. I think Professor Rao knows that. The project had started, but then they required a nodal pathologist to kind of put things together. And so they approached me at NIMHANS and I willingly agreed to get into the project. And that's where I met all these fantastic people, which I will cherish forever and ever. I had very good clinical colleagues. Dr. Chandramoli is here with me today, Dr. Anand, with whom I worked for several years. They taught me so much about the clinical aspects of all these brain tumors, and they gave me a different perspective about research also. I have a special relationship with Arivalgan. I think everybody knows that. He's worked with me for several years, and we not only in this project, several other. He's also a pillar for the neuro-oncology lab. Coming from neurosurgery, his involvement in neuro-oncology lab has been superlative. And that's how the neuro-oncology lab also, I always got confidence when with Dr. Arivalgan with me, and we even continue to collaborate. Till now, we are collaborating with each other. And these are some of the collaborative work that we did, which has already been shown, and the tissue microarray technique. I'll tell you, it was so difficult to get this tissue microarray equipment. At that time, we searched all over the world to get one equipment. There were only 2 or 3 companies making it. And there's only one from Beecher company that I was able to get. It's a semi-automated one. And when I bought it, the vendor told me, no, you don't need any sales or service for this. Nothing will happen to it. And it's so many years now and nothing has happened to it. So that's how I enjoyed doing this tissue microarray technology. My technicians, they are Mr. Chandrasekhar, who's seen over here. He's really an expert in this TMA technology. And I've enjoyed guiding several students, their PhD students, not only from our department, from other departments as well, including clinical neurosciences. DM neuropathology, PDFs, and even MCH neurosurgery. I used to have at least 2 students every year working with me in some neuro-oncology project. And I think what I can say is that it's the dedicated work of all these students that have helped me to achieve my goals. And that is the neuro-oncology lab. And it's a central facility, of course, and we open it up for all other departments and started the self-sustaining diagnostic molecular neuro-oncology facility as well. And these are just some of the papers that have recently been published from my PhD scholars in the department. And I'm very proud of each one of them. The amount of work that they have put in and the amount that they've taught me is something that I can never forget. And my contribution to the Indian Society of Neuro-Oncology, this goes a long way. They were all showing this as my contribution, but let me tell you, this is a group effort. And it was spearheaded by Dr. Rakesh Jalali and Dr. Chitra Sarkar. And it was so wonderful working with the group and bringing out the algorithm for our country. And now we continue to do that for the Asia-Oceanian region. So my neuro-oncology team, once again, I want to tell you that Arivalgan is a big part of our team. Yasha Nandish worked with us in our previous projects, and Shilpa, I have a special mention about Shilpa because when she joined DM in neuropathology, she, with a lot of interest, she came to me to do her thesis work. And we did some kind of a molecular work on glioblastomas, which she took it forward. And then I could see how much interest she has in molecular neuro-oncology. And then she got into the lab. She's proficient in almost every technique that is there in the laboratory. And now as a faculty, she's able to amalgamate that with histopathological diagnosis. I'm sure she's going to take it to greater heights. I just want to talk about few of my students, Balram, Dr. Arun, Lieutenant Colonel Rajmohan, Kritika, Shravya, Vidya, Harsha, Raju, all of them are my PhD students. Harsha is my last, she's the baby of the team. She's my last student. She's just about to submit her thesis before I leave this institute. I hope she does that. And then I had DM students also working with me. We had Sumitra, we had Radhika, we had Sautrik. They all did neuro-oncology work. And of course, we had project assistants, Nandaki, Janavi. And definitely, I want to mention my technical staff, Chandru and Suresh. Chandrasekhar is the one who is an expert in the fluorescent in situ hybridization technique. In fact, there are so many, he gets phone calls every day from pathologists, technicians from all over the country for troubleshooting. So that is his expertise. And last but not the least, I want to mention Anantalakshmi. She has been with me for many years, more than 15 years she has been with me. And she joined the project, she worked in the project, did admin work for all my projects, starting from NIMHANS downwards, and then got into the self-sustaining lab and has done excellent work in the neuro-oncology lab as well as in the self-sustaining laboratory. So I'm very fond of all my students and the project assistants and everybody who's in the neuro-oncology lab. I think it's all because of you that I am here in this place. And now we are all experts in all techniques in the department. And recently we had a molecular neuro-oncology workshop. It was spearheaded by Dr. Shilpa, with ably assisted by all the members of the neuro-oncology team. And we had about 10 pathologists and neuropathologists who came from different parts of the country to learn the hands-on technique so that they can start that in their departments. We had EM workshop, and Dr. Anita had spearheaded very nicely an immunohistochemistry as well as a special stain webinars, which was conducted so beautifully by our technical staff in the department. We are very proud of that. I am very proud of that. And my faculty members, I mean, I don't consider them just as my co-faculty members. They're all such friends to me. And as you can see, we are dwindling in numbers. First, Dr. Gayatri retired, then Dr. Sagar. Now I am going to superannuate. So I'm sure there'll be a new breed of young neuropathologists who are going to step in and take the department to greater heights. I've had the good opportunity to work in other departments as well. And I was the in-charge head for the clinical neurosciences for almost 5 years. I think it was 4 to 5 years that I was heading this department. And I've had a very good interaction with my other faculty, Dr. John, he's here today, Dr. Ravi Yadav. I think I can never forget all the discussions we had during these days, and the wonderful set of young clinician researchers who joined the department. They were with me for almost 4 years, all of them. Young, brilliant minds. They have finished their PhD in different aspects of mental health and neuroscience. They are settled in various parts of the country as well as the world. And I even got a chance to head the transfusion medicine and hematology after Dr. Sundar Periyavan superannuated, and was very fortunate. I've been very fortunate to have brilliant youngsters, Dr. Vijay Kumar, Dr. Param Tripathi, who are there here today, Dr. Sangeeta, Dr. Malita, the technical staff, the secretarial staff, as well as the residents. It's been a wonderful journey for me in transfusion medicine as well. I've cherished friendship which is everlasting for me, inside the department, within department, outside the department. And within the department, I can tell you that I've had very close friendship with Yasha and Gayatri. We are buddies for lunch buddies for more than 2 decades, and I will cherish that friendship forever and ever. The other faculty, Nandish, you're not there in this photo, please excuse me, but the other faculty, they're not just my other faculty members, they're very close friends to me. Anita, Shilpa, Nandish, Sagar, Rashmi, all of them extremely friendly. That feeling of a family is always there when I'm in the department. Friends from outside, and Dr. Ravi, he is not just a friend, he's like one older brother to me. Always comes to the department teasing me, but giving very sound advice. This was during COVID times. We used to call him COVID Baba. And that's how he was. And we had friendship even when I was as a resident or, early in my career, friends from other departments. I don't know if you can recognize Dr. Saroja, Dr. Bharati. They're all senior neurologists, well placed in the country now. And my everlasting friendship with Shantala, which I don't think I can ever forget that. And of course, friends from outside, from the neuropathology group. You can see the famous ladies of neuropathology over there. And they're all such close friends to me. And I've shared so much with each one of them, Chitra, particularly with Chitra for many, many years. And she's not just a mentor, but, she's a very close friend and very warm person that I have been associated with. Geeta, Kirti, Vaishali, and others. Not only our neuropathology, from other disciplines also. You can see Anand, you can see Sandeep Chatterjee, Rakesh has been one of my very, very good friends for so many, many years, actually. And last but not the least, my family. I'm always indebted to my husband, Dr. Santosh. He's here today with us. And we've been married for so long. He's been my long-standing companion, but let me tell you, he's my best friend. And I can share anything and everything with me. And he has encouraged me and supported me throughout my tenure here. My son, Nikhil, and my daughter-in-law, Brandy, they're like pillars to both of us in every aspect of life. And they're always there for us. So I just love them. The elders in my family, my in-laws and my parents. This is my sister-in-law, Dr. Lata, and she's Santosh's younger sister. And her husband, Dr. Vijayaram, they are extremely supportive to our family, to me and to Santosh. And of course, my siblings, that's my older brother, Amarnath, my sisters, Prabha and Sadhna. I share a lot with my siblings as well. So it's been an incredible journey till now when I'm going to step into the next phase of my life. And I think this journey has been incredible because of the everlasting friendships that I have fostered over the years. And the excellent work environment. That is one thing that I can say, that this institute gives you excellent work environment. And we have combined work with fun times. And what more can one ask for beyond that? And I'd like to thank so many colleagues, so many friends, not only within NIMHANS, but outside of NIMHANS. They've supported and helped me in ways that are too numerous for me to recount. And I'd like to thank all our admin persons. First of all, our past and present directors, registrars, deans, MS, RMO. Without their support, we could have never done any of this. We could not have even expanded the department. All the other departments, the administrative staff of neuropathology. I'm ever indebted to all the staff of the neuropathology, as well as transfusion medicine and hematology. And lastly, I think I'm still working on finding the best in me. To me, it feels like it's an exciting ongoing journey. I don't know when that is going to end, but it is going to go on and on. And I'd like to thank you all for being my traveling companions. It has been a journey. So I would like to thank you for being those traveling companions along a very large and important part of my life. And the enormous collection of shared experiences with each one of you, this is permanently imprinted in my mind. Thank you so much.
Thank you so much, madam. It has been a mesmerizing talk, very inspiring and very encouraging. And it's so nice to listen to you, feel like rewinding and listening again. And all of you will agree with the eloquent of words madam used, like an extempore it came. And I'm sure the madam's talk has been refreshing. It has filled our stomach, but still formality sake, we may need some tea and break. So we'll have a very short break and come back for the next part of the program, that is reminiscence and sharing of thoughts and words. Okay, we'll have a short tea break. 10 minutes. 10 minutes, yeah. Yes. 10 minutes. welcome back. Madam has a very magnetic personality. Anybody who comes in contact, interacts, or start by Madam will have some kind of warm memories which will be carried with them. So I'm sure many would like to express their thoughts about Madam, their experiences. There are many guests who are online. So we'd like to start with the guests who are online. I would like to start with Dr. Sarala Das, senior neuropathologist, former head and professor, mentor of Madam Vani Madam, to express her views and share her thoughts.
Hello, are we audible?
Your audible voice is little feeble, Karishka sir.
Okay. Is it better now?
Little.
Okay. Well, let me convey what my mother, Dr. Sarala Das, has to say.
Sure, sir.
Yeah, Doctor, she has written it down for me to read it.
Yes.
Dr. Vani was one of our early senior residents when our department was in the formative days. She has risen to great heights in neuro-oncology and has kept our flag, NIMHANS Neuropathology Department flag, flying very, very high. My blessings to Vani on her superannuation. I am sure she will continue the good work further. Best wishes to you, Vani. So this is what she has written down, and she remembers very fondly the personal and professional moments that they have shared over the years there at Bangalore. And she recollects Dr. Vani being an extremely helpful person on all fronts, both professional and personal, and wishes her very well for the future. Thank you.
Thank you, madam. Thank you, Kanishka sir, for that. I now request Dr. Asha, another close friend and senior of Vani Madam, former colleague of Vani Madam, to share her thoughts. Okay. Ravi, I request Dr. Ravi, former registrar, dean, and HOD of neurovirology, to share his thoughts. Sir, you are muted. Muted.
Can you hear me now?
Yes, sir. Yes.
Good evening, everyone, and my profound apologies for not being there personally. I've been held up with some COVID-related issues since morning. It's indeed a great occasion where we are celebrating the academic and clinical services rendered by my very dear friend, Dr. Vani Santosh. I've known her for 35 years. She, of course, is internationally renowned for her contributions to neuro-oncology and various other aspects of pathology. I've had the privilege of collaborating with her in many, many projects. And I would like to thank her for the special affection she showed towards me, more so during the difficult times I had with my health 3 years back. She was a great source of support. Vani is a very, very fun-loving person. I don't know how many of you know it. And she has been unfortunately subjected to a lot of my pranks. And I just want to narrate one, because this is a fun incident. When I was a registrar, Dr. Vani was HOD neuropathology, and she asked for buying a vibration-free table for their discussion microscope. and it was estimated at about 1.5 lakhs. And I asked her, why do you need it? She said, no, all of us have to strain our necks and so on. I told her, I'll give you a simple solution. I'll give you 10 tins of Complan. All of you drink, become tall. Or I will put some bricks under the existing table so that they would not have to strain their necks. And after that, all hell broke loose, and I was thrashed black and blue by Vani and her friends. So that's the kind of close relationship I have had with the pathology department, including Dr. Vani. Vani, it's a great occasion. Welcome to the Retirees Club. And let me tell you that you will get more busier than what you are. And you will have great opportunities to contribute. Thank you, Anita, for giving me this opportunity. Thank you.
Thank you, sir. Thank you. I now request Dr. Anand, Senior Consultant Neurosurgeon, former Professor of Neurosurgery at NIMHANS, to speak a few words.
Thank you very much, and it's a wonderful opportunity. Thanks for the privilege and honor. It's been a great privilege and honor to know Dr. Vani since I joined NIMHANS way back in 1993, and we have known her professionally as a student, then as a colleague in the department, and as a collaborator later. And she's, as a person, very warm, very affectionate, and as she said, she had treated all of us from student days itself as part of family. Her knowledge in neuro-oncology is practically unparalleled, that I have not seen with anybody else. She's so curious, she's so passionate about her work that she will know even before the days of internet, an article which is published, and she would have read through it and she would have digested the information, and she will be raring to go, when are we likely to do it? And she used to be full of questions, curious to know what are the clinical implications, why this tumor is behaving like this, how do we answer it? And she was always very happy to guide students. And we have had very good moments in collaborating, starting off the glioma project, and the success of all of them has been her encouragement, the love and affection she has shown to each and everyone. As she said in her talk, she considers the whole place as family. And her passion is so infective, and perfection. She will write the paper herself if she is not happy with the student having written it. And that amount of dedication, time and again for over 30 years, just amazing to see in an individual. And that amount of energy she brings in, she's a constant source of inspiration for all of us. And every one of her talks on the same subject, even if you listen after a month, you will find a new slide, you will find a new information, which she has pulled out from somewhere, which even you cannot read, even if you search the internet. And she will tell you in proper perspective, whether that's essential, if it's good. She would have spoken with the author, or she would have corresponded with them. And the way she has set up the lab just shows how humbly and modestly she has started off, without any great ambition, but purely by passionate work and dedication and commitment. She has achieved great heights in neuro-oncology and helped everyone in the journey rise up to their excellent potential levels. And it has been great knowing her family, a wonderful husband, supportive, and a lovely son that they have brought up. And I'm so fortunate to be part of them and know them as family. And thanks, Dr. Anita, for organizing this, and to the Department of Neuropathology. And I'm sure she has a lot more to contribute, as Dr. Ravi said, in a more meaningful and consultative manner than having the burden of service post-retirement. Thank you so much, and thanks for the opportunity.
Thank you, sir, for those nice words. Now, request Dr. Sagar, his former colleague and professor of neuropathology, to say a few words.
Am I audible?
Yes, sir.
Good evening. Sorry, I am not able to attend these functions as I'm in remote area of Shimoga. Today, it's a proud feeling for me and an honor for me to say a few words about Dr. Vani Santosh. Dr. Vani Santosh is more than an elder sister to me, though I am elder to her officially. But biologically, I am 6 months younger to her. I have been associating with her since she joined the neuropathology department. What I found with Dr. Vani, she is highly dedicated and committed person. Her contribution to neuropathology in diagnostic research is enormous and internationally recognized. She is highly committed, a composed and balanced lady. She is a person who never hurt anyone. She has been always respecting individuals' feelings. When it comes to her administration, she is very, very hard and strict. It is very difficult to convince her, but once she convinced, she can go to any level to get things done. During her HOD-ship, we have got many useful instruments to EM lab. She's always helping people, both academically and personally. She's one of the person who encouraged me in academic and professional development. I had a great time while serving the institute with her. From my bottom of heart, I wish her happy, healthy, peaceful new life. Jai Hind.
Thank you, sir. Now I request Dr. Vaishali Suri, Professor of Pathology from Aryan Institute of Medical Sciences, New Delhi, to say a few words.
Hello, can you hear me?
Yes, madam.
Yeah. I would just like to summarize what others have said. मेरे लिए ये बड़े गौरव की बात है कि वानी मैडम को मैं भी अपनी श्रद्धा अर्पित करूं। इंटरनेशनल लेवल की इस महान संस्था में उनका कंट्रीब्यूशन अमूल्य है। उनकी त्याग तपस्या से न्यूरोपैथोलॉजी का गौरव बढ़ा है और उनके प्रति मैं कहना चाहूंगी, जहां कहीं है ज्योति जगत में, जहां कहीं उजाला, वहां खड़ा है कोई अंतिम मोल चुकाने वाला। मैडम, wherever there is light, there is one person who's sacrificed everything, and that is you. And उनके व्यक्तित्व के कृतित्व में कवि की यह पंक्तियां बहुत ही सार्थक है। जिनकी आंख में है स्नेह, जिनका शुद्ध उज्जवल भाल, जिसके अधर पर मुस्कान, जिसका हृदय सिंधु विशाल, सुंदर स्वस्थ प्रतिभावान, जिसके हैं महान विचार, जिसके कर्म गौरवपूर्ण, जिसका विश्व है परिवार, जो कर्तव्य तत्पर नित्य है, तप तेज बल श्रीयुक्त, सेवा सत्य सुख का रूप, दिव्य दिनेश जीवन मुक्त, दिव्य दिनेश जीवन मुक्त. You are an epitome of love, an ever-smiling, pure soul with heart of an ocean, unmatched talent, a person with great thoughts, whose deeds are glorious, for whom the whole world is one family. And you are a strong, beautiful lady who served humanity all her life, and you'll be continuing to do that, madam. You are an angel with a liberated soul. Thank you so much, and thank you for being part of our lives. And thanks to Dr. Anita to give me an opportunity in this evening. Thank you.
Thank you very much, madam. Now we'll go on to the members who are on site here. We'll start with Dr. Chandramouli, Senior Consultant Neurosurgeon, former Professor and Head of Department of Neurosurgery, NIMHANS.
Oh, I'm not a very good speaker. Kindly excuse me. We have been having a very long association, as all of you know, that Dr. Vani and us. We are not only very well connected or emotionally connected with neuropathology department, not only Dr. Vani and the entire department. Every year, even Vani has to do, you have to come here and show that we are alive and kicking. So I always say make it a point to visit Department of Neuropathology and meet all of them. So what I've seen her not getting angry at any point of time. She's always cool, composed, and I've never seen her fighting with anybody so far. That is her character. And all the residents, at least in neurosurgery, would be running to her to get a topic for thesis. Simple thing is, their job is just to give the samples and forget. Rest of the things will be done by her. That is the simplest of the thesis that I can do. So she has been a good human being, basically, I would say. Of course, all the professional achievements, everything has been highlighted. Of course, she's a very jovial person. Like Ravi, we also used to occasionally pull her legs. She used to take it very sportively and we used to have nice interactions. I'm sure she's being pulled apart now by many organizations or individuals to join. I think she's going to make a very good choice and continue to serve the science as well as the humanity. Thank you.
Thank you, sir. I request Dr. Vasudev Rao to say a few words, share his thoughts. Senior consultant pathologist from PGS Global.
Just want to tease me. Wait a minute. He will tease me.
Excuse me. Thanks for giving me this opportunity, because every time somebody leaves the department, I'm invited. Sad part. Then last part is, he said I'm a senior consultant at some hospital. I'm not.
Sorry, sir.
I am actually, I spend most of the time in the kitchen, or I'm eating, or feeding the dogs and the cat at home. Now, all the while you have listened to Vani post NIMHANS and further. Nobody in this audience, including Santosh, including him, would know about what I'm going to tell you.
Oh God, I know it's embarrass me.
I don't want to call her by any prefix or a suffix. I just want to say Vani. Even now I'll call her Vani, because I know her going to school. I know her, and I know her. I bet only she will understand. Cycling with a tennis racket stuck to the cycle. See, there are two things here. Now, knowing a person, somebody may ask you, do you know the Prime Minister of India? I say, I know him. Does he know you? I said, no, he doesn't know me. I know him. Likewise, Vani did not know that I knew her. I can stand here. Okay, now that's the funny part. Now, for a long time, she did not know, because I knew her brother-in-law since childhood, and her sister also. And then we knew that Vani was there, and we would see her cycling. Then I came to know that Vani was in the medical college. Still, I did not know her. Then I knew that she was doing her MD pathology. I did not know. But I knew everything about her, but she didn't know me. First time I saw her after a long time was when she came as a MD student to the Department of Pathology to learn a little bit of neuropathology. She came with Shamim, I'm correct. And I can clearly remember how she went into that room in the museum. We had kept a lot of these brains. And I told her, this is what, this is what, this is what. And then they all went away. With this, she got inspired at that moment to see more brains. I don't know. Then after that, I went off to Paris for my postdoctoral. That's in 89. Then Shankar probably wrote to me saying that Vani has joined as senior resident, 1989. So that's when I came back and she was resident. Then, of course, after that, you've heard everything about her. So which means that I don't have to call her by Dr. D, HD, and all that stuff. I could call you Vani, okay? And after that, we had a very short, brief period together at NIMHANS because I was planning to go away. That's 89, 90, 91, 90. 94, I left NIMHANS. But I still remember everything in the department and how we used to interact for those 4 years, teaching her how to cut brains and seeing slides together, making diagnosis. Those are all days of morphology, you know. See that fiber there, see that fiber there. Where is the fiber? Oh, the glial fiber. Ah, this is a glioma. Look at the nucleus, that's all, not beyond that. Immunohistochemistry, we never knew anything about it. Computers were not there. Mobiles were not there. We have to go searching to the library, take out books from there in the dust, and with all that we would read a little bit of immunohistochemistry or just then coming up. If you do GFAP, that was something great, you know. So after that, I've been following up the department very closely. Now and then I come, but I remember what was happening in the department, and all praises to you. Second is another thing. Now that you're beyond the demands, I want you to continue to cycle. Now this is an inverted commas. You need not have to cycle. You have to go slowly. See, that's very important. All these days you come in a car with a driver or you're driving, you don't see anything on either side of the road. Familiar roads, you don't know the shops. A good example is that when I was going to my hospital these days, every day in the car and all, you don't see anything. So I gave my car for a servicing, I would take a bus. And the wonderful things you would see which you had not seen earlier. There was a big wall and I wanted to see what's there inside. Every time there's a wall and the gate, I would see. When I went into the bus, you relax, you see on either side. And there's a beautiful playground inside. There are a lot of flowers and all growing there, etc. So which means to say, now you come slowly, take it relaxed, see on either side, see again prostate, see an endometrium, see cervix. You will enjoy, Like going to a flower show, you grow some flowers at home, but you go to the Lal Bagh show to see different flowers. You know? Beautiful. Pathology and the other organs are also very nice. You see a liver. That's what I used to do. You know, after seeing all these brains, I'll go somewhere and see some slides. Very helpful for me. And special stains. You know, Kumar, fantastic. Is he there or he's gone back? He's not there. Now we used to do special stains, you know, and that fat stain is so important and a lot of people did not know. And it helped me enormously. I used to do a lot of transplant pathology, the liver these days. I did a lot of liver transplants and middle of the night. Like middle of the night story is there was no phone, there's no mobile. Suddenly at 2 o'clock in the middle of the night, there'll be a big buzz, bell. You open the window, money. Vishwambaram is there. Oh, God. Sir, autopsy there, sir, money. 2 o'clock and all I've done autopsies. One day it's biliary tuberculosis, full of. They were all flying out. Vaseline. When I did the autopsy, I went off. So this fat globule, they called me at night for liver transplant. If you want to give away the body, they'll want to know, take off the liver before morning, and they want to give the body, and they'll make a biopsy and frozen section to assess the fat globules in it. The surgeons thought they knew everything. They came and said, this is no use, 80% of fat in this. This is not fit for transplant. Then I would tell my technician, you put it under the microscope and then call me. So under the microscope, I went there. All the fat globules were floating on top. The liver was fantastic. So they did a transplant. So what I meant to say is, around, go slowly in life, take your family, treat them well, spend more time with them. Pathology is there. It's always there in your mind. Any day you can do. What you did not do all these days, please try to do all those things. You'll have a wonderful life. You've got wonderful husband and family. God bless you all. Take it easy. Keep in touch with the department. Come now and then. I've given a challenge to the department. 117 bar 1979, 117. I don't think you all took it up seriously. Tell me what that autopsy is sometime. It's an autopsy on a dog. Okay, with these few words, I keep talking about department. I'll never stop. God bless you all. And please keep calling me, not just for retirement, but anything else also. If I'm free, I'll do come.
Thank you, sir. Now I request Gayatri Madam to share her thoughts and say a few words.
So good evening, everybody. You have heard so much about Dr. Vani, so I will put in my thoughts, and then I will also show you some of those photographs which we have asked her to give it to us to share it with you all. You heard about the academics of Dr. Vani. Now, it is my great pleasure to share my thoughts about Vani, with whom I was associated 1989 when she joined as a senior resident. You know, Vani topped in the MBBS. She got gold medal. I don't think anybody know about it. She got the gold medal. She not only got the gold medal, now she is also a tennis star that has been told, and a sitarist. So you can see the photographs. I think the students were in awe with seeing all these. So now you can see that here. Actually, these are the slides which Dr. Yesha was supposed to present, but then she gave it to me. So here is Vani with the team, and you can see the paper advertisement that was given that she is going to meet with Jaya in the final. I don't know what happened in the final, but anyway, she met Jaya in the final. And then here I see this photograph. She's being congratulated by somebody. I thought she won this match. So congratulations to you, Vani, on that fate. She's a sitarist, and Vani, I think you should now continue to do this. So you know how much you love music, and she's a Hindustani sitarist, so she should continue to do this. So I told you my journey began in 1989 when she joined the department as a senior resident, and I was just a research scholar from a basic science background, and then I don't know anything about neuropathology. Thanks to Dr. Vasudev Rao, thanks to Madam Sarla Das, thanks to Dr. Vani, thanks to Dr. Asha also, who taught me neuropathology. But most of the time that I have spent in the lab seeing the slides with Dr. Vani and Dr. Vasudev Rao. So we had this nice double head microscope, and we used to play musical chair. Vani will sit first, she'll finish seeing, and then I have to take in that chair. So, and later we used to discuss about the findings. So that's how I learned neuropathology from both Dr. Vani as well as Dr. Vasudev Rao. And of course, Madam Salladas was the mentor behind whatever I have achieved in muscle pathology. So apart from learning and seeing the slides, it was the autopsies. She told you that we had about 300, 400 autopsies per year. And then every autopsy I used to accompany her. The reason being she was so interested to take all the tissues for electron microscopy. She had a passion for electron microscopy. And hence, okay, before I go into that, I think this is the family photograph. I think I did some jugglery, and this is the family photograph. You can see her beloved husband, and these are her sisters. Her elder sister Prabha. I hope I'm right, Vani. She's the elder sister Prabha. She's Sadhana, and this is Aarti, your younger sister. But I didn't have Ammi's photograph. And these are her friends, and one of them, I think some of you can recognize. She's Ushakini, right? Yeah, she's Ushakini here. And then her beloved son, Nikhil. So having passion for seeing what is happening to all these tissues at electron microscopic level, we took a lot of tissues, including liver. Sir, we have taken liver also. Liver, kidney, the various regions of the brain we have taken. And like Dr. Yasha has put in, her interest was not just neuro-oncology, but it was peripheral neuropathy initially. And I have learned peripheral neuropathy from her, and then infections of central nervous system. So she wanted to see everything under electron microscope. And I must tell you that we have spent such quality time seeing all these grids under electron microscope. And we had some vague cases, and then we did the diagnosis, and we were so proud of ourselves for making the diagnosis. I do not know whether the diagnosis is right today, but then we did. So working with a colleague like her was so much fun. So these are the photographs. So because she wanted to do electron microscopy, we had an electron microscopy workshop, and then she registered for the workshop. And here she gets her certificate after having attended the workshop. Her interest was also in skeletal muscle. So this is the workshop that we conducted, and you can see her here sitting and listening very carefully for all the lectures that we had on skeletal muscle disorders. So apart from this, I must tell you that during my thesis time, when I have to defend my PhD thesis, she's played a very important role in me. So we had those projection slides. I don't know how many of you know the projection slides. So we had the projection slides, which had to be loaded into the casserole and then projected, and then we have to see. So initially it was single projection, and then it was double projection. So when I had to defend my thesis, I had a huge number of slides. And she said, no, how are you going to do it? So let us have double projection. So we said, okay, double projection. The first time I have been using double projection. She said, you know how to use it? I said, I don't know. I know only single projection. Then she sat with me. She numbered each and every slide, left, right. So slide number, I still have those slides. Slide number 1, left, slide number 2, right. So this is how we did. And then she made me rehearse. We sat till 7 o'clock in the night to rehearse my PhD thesis work. And then the next day was the presentation. So I thank you very much, Vani, for that. And apart from that, our conferences. So we have attended almost all the conferences, NSI, that is Neurological Society of India, and the Indian Association of Pathologists and Microbiologists. We must present papers in that. That's one of the goal of the department. So both of us used to go to all the conferences, and our journey to the conference, it is a pre-conference journey, then you have the conference, and then you have the post-conference. So the pre-conference journey was in the trains, the second class we used to travel. It used to be full of NIMHANS residents and then the PhD scholars. So all along, including Dr. Ravi and Asha, all of us used to go. And when all of us in the department used to go, it was only Dr. Shankar who used to hold the department. He used to let all of us go, and then he'll say, you all go, and then I'll be there in the department. So along the journey, it's not eating, it's not enjoying. We have to sit and rehearse. So she will rehearse, I have to rehearse. So it is learning even in the journey. So after the conference, of course, she has won awards and I have won awards because we did such a wonderful work. Post-conference, we used to sneak out. So here you can see Vani, myself, and the other pathologists. This is my daughter. We were in IIPM Cuttack, so we had wonderful time in Cuttack, and Madam was very particular that we see. They are very proud of their state. They said, no, you must go around and see. And this is the Konark temple. We stood by the Konark temple, and then we have taken this photograph. You can also see Dr. Saubhagya here. So that is the IAPM in Cuttack that was held. And this is the NISI conference in Jaipur. So you can see Dr. Ravi here, then Dr. Anita, then Vani here, and of course, Dr. Chandramukhi, Anita, all of us. And this is Ratnavali, the neurologist, who was a faculty here at NIMHANS. So her support was extensive during this journey of my thesis, as well as all the presentations that we made in the conferences. People have talked about her administrative capacity, and as a HOD, she did a great job that everybody has spoken about. So her contributions and her dedication will always be the prime example for others in the department. One thing which inspired me is her decision-making skills. So that was, she was quick, and if she's not able to make any quick decision, she'll say, you just give me some time. And then she comes out with an answer. So that it has always been inspiring to me, Vani. So I thank you for being at my side, or by my side, during all the hectic days. And I truly appreciate everything you did for me over the years. The one thing that I want to tell on a personal note is, in addition to being a colleague, she's a very great friend of mine. And she's also a part of our family. So she was at my side when I fell sick. And I just gave her a call, and then she came home. She took me in her car, and then straight she took me to Amarjyoti Nursing Home, where Dr. Santosh was practicing, and admitted me there. So both Dr. Santosh and Dr. Vani has taken care of me during those days when I have fallen sick. Thank you both, Dr. Vani and Dr. Santosh. I was admitted for 3 days, and then I came out of the hospital, and I'm alive and in front of you now. So she has told us about the fun times that we used to have in the department, the famous picnics that we used to have, and also the parties that we have in the department. It started right in the 1980s when I joined, that we had parties, and Dr. Rao is also one of them who loves eating. And then we used to have parties every other month, to the extent that the neurology department at that point of time was just below us. There was the OPD there, and then you have the neurology faculty rooms also there. One Dr. Arunodaya comes up and he says, I think your department is an extension of canteen, because we get that nice smell to the department. So he always used to joke with us, and then he will barge in and then have with us, eat with us, and then go. So that is how we have enjoyed our stay at NIMHANS. So this is the Bannerghatta outing that we had. You can see Vani here playing with the sun. So this picnics were for entire family, including the spouses, but spouses never came with us. It was only the kids that came with us. So the spouses were away. So we had fun time in the picnics. And then, of course, we always had parties. You can see Nikhil over here, Vani here. This is Dr. Saubhagya. This is Preeti Ram Mohan, Dr. Ram Mohan's daughter, and this is my daughter. So we had
We had a lot of fun during those days. So now comes the time that we spent together, and then we had fun time shopping for both her son's wedding and my daughter's wedding. The reason I put up this is, so my daughter got married in January and Nikhil got married in the month of March. So when they came for the wedding, she was so happy after everything. She hugged me, and then, you know, she wouldn't leave me at all. So I thought I should put this photograph. And I also put this photograph because you can see Dr. Santosh here. Are you able to see that? So immediately after the wedding ceremony was all over, he came up to me and he said, Nikhil is getting married next. I said, yes, I know. We shocked for them. He said, the peta will come to me for Nikhil. So whatever peta that we put on Kevin, who is my son-in-law, went to Nikhil. So Nikhil has the same peta. So that's the friendship that we had. And, you know, they are the memories that I will cherish forever. So Vani, as you embark on this new path, I wish you good luck, have good health, a happy and peaceful life with your family. On a personal note, this is the family time that she had recently, I think. This is the entire family, Dr. Vani, Vani's family, and this is Lata and Vijay Ram, that is Santosh's sister and brother-in-law. Here you can see Nikhil. That was a baby photograph that I showed. Here you have Nikhil, and then you have Nikhil and Brandy here with the mom hugging. So I wish you all the best. On a personal note, I would say that Vijay, Ami, and Kevin also send their best wishes to you and to the entire family. So we have had good time. We did sneak out only twice over this 3 decades of our stay at NIMHANS, just twice. The 3 of us, we sneaked out, and then we thought we should be kids again, and then we ate some burgers, and then we came back. So this is during my retirement. So Vani, I welcome you to this retirees club, while Yasha has to wait for some more years. Thank you so much.
You're still holding the mic?
She wants to speak next. Thank you so much. Thank you so much.
Anybody else from the audience want to speak? Yeah. Dr. Shanta Legde from Department of Clinical Psychology, additional professor.
Shantala will sing the song for her?
Yeah, at the end.
Okay.
But I need to say a few words before that. I just got this acronym. Everybody is using acronym. I know sounds a bit silly, but still I will say that. It's like BMW, no? Very classy, like Vani ma'am all the time. But B for brain, that's how my connection with neuropathology department actually started. I was so passionate about learning the anatomy, brain, and as a student, Shankar sir was taking classes. So I used to come to pathology department from the ground floor where we had the neuropsychology lab. So Sundays, half a day would be neuropathology. And that's when actually I met Anita, who almost like was the elder sister, you know, with Shankar sir, and picking up the brain from the bucket and learning the anatomy. And that's how I think the brain connection happened. M was music. I think the music connected us. I remember after my PhD, and just I was about to finish PhD. I mean, after finished PhD and just about working in the area of music and cognition, and I was SSO. We had the music lab, pathology lab, and I think Dr. Venkat's lab in that building, huge NBRC and nothing else. And above all dusty sometimes. It's just 3 labs, and we used to often have VIP visitors. Every other day we used to wait for some VIPs to come. And then everybody would want to know about music research. And the W part of the BMW was the 3 women I always admire. Yasha ma'am, Vani ma'am, and Gayatri ma'am. The 3 would come to me, and all 3 would adore me. I think, yeah, sometimes Yasha ma'am would say, you have special love for Vani. I think they would, she would say, you have special love for her. And I say, ma'am, don't feel jealous. I love all of you. But yes, that's true. I do have special, I don't know, sometimes you just connect, there's no reason as to why, but all 3 of them would make me sing or say something. Oh, you're doing such good research. I mean, they would just want to know so genuinely as to what work I'm doing. I was still starting my journey. And here were 3 women who I knew had achieved so much, and I know much more today about Vani ma'am than I knew really, truly, about all the glioblastoma work and everything. But so that was the BMW. And I always, whenever I would visit pathology department, it was Shankar sir who treated me like a young kid, and a daughter, Anita, who was like an elder sister. And all 3 of you treated me like a daughter. And I would always admire the connection that all of you had like a family. I think that's what I think everybody would maybe envy and, would want to emulate from all of you as a department. And especially me, although neuropsychology, and now I'm indirectly connected through Arivalagan to do some work on psychological aspects and, oncology patients. But although I did not work with you formally, there would not be a direct connection. I think music connected us. And I'm really fortunate to have made this connection in this place. And NIMHANS, I'm truly grateful for giving me like this opportunity to connect with souls who mean a lot beyond NIMHANS. So when Vani ma'am said she's retiring, I said, this connection will not go. You'll go from NIMHANS, but I'll always be in touch with you. So she's a dear friend. I've had my lows, challenging times, and I think everybody has said that's always smiling. Just one hug from her, you would say, calm down. Now it's okay. Everything will be fine. And always so positive. She would say, don't worry, everything will be fine. Everything will be fine. And I would often visit, and Ananta Lakshmi would always know, you have come, so you can go in now. Although she was busy, I would sneak in and say, take some time and talk to Vani ma'am. And she, not just that, another part of her is she would always inquire, how is your work going? Be focused. How many publications are you doing? Keep this as your target. So she was not just loving, caring, but also I could see that strict mother side of her, which I would love. And I think implicitly, I've learned so much from you as a person. And yeah, lots of love to you and to your family. And the journey is just beginning. So you're still young, as you said. Number is 65 and all is just a number. So lots of love to you. I don't know what to sing for you, but
Something, one song.
Just a line. Just a line maybe, because I feel this journey is like a ship and we all have a long journey to go and it's ups and downs, but ये हौसला कैसे झुके रुके, ये हौसला कैसे झुके, मंजिल मुश्किल तो क्या, धुंधला साहिल तो क्या, तन्हाई दिल तो क्या हो, ये हौसला कैसे रुके, ये आरज़ू कैसे झुके, मंजिल मुश्किल तो क्या, धुंधला साहिल तो क्या, तन्हाए दिल तो क्या हो.
Thank you, Dr. Shantala, for that wonderful song and words. Dr. Kumar Velu Somasundaram could not attend this program, but he has sent a message. I'll read on his behalf. I'll read verbatim, not miss any word. I want to share my time spent with and interactions with Professor Vani Santosh. I have known Professor Vani Santosh for the past 18 years. I remember meeting Professor Vani Santosh for the first time when she visited Manipal Hospital, located on the old airport road. I was part of the CSIR NIMHANS project 2000 to 2004 headed by Professor MRS Rao. This project started in 2000 with the mandate of transcription profiling of 3 cancers, that is glioma, oral cancer, and gallbladder cancer. Professor A.S. Hegde and Sridevi Hegde were PIs from Manipal Hospital. We had some difficulty in grading glioma. We were told that if there was a person who could help us, that is Professor Vani Santosh. Professor Vani Santosh was invited to a meeting in Manipal Hospital to discuss glioma classification sometime in the year 2004. The first paper, that is upregulation of ASCL1 and inhibition of Notch signaling pathway, characterized progressive astrocytoma, with Professor Vani Santosh as a co-senior author, was published in the journal Oncogene in the year 2005. Characterizing progressive astrocytoma could not have been possible without the contribution of Professor Vani Santosh. This paper has been cited 150 times so far. This is how our journey of working together on glioma started, and we have now co-authored at least 45 papers. Although we started with the idea of investigating 3 cancers in the CSIR NIMHANS project, the glioma work moved fast, and ultimately we could do a lot of work during the last 15 years. My laboratory at IISc has been working on the genetics of gliomas for the past 22 years. I can certainly say for sure that the participation of Dr. Vani played an essential role in pushing the work on glioma to a greater level. We had the second CSIR NIMHANS project 2005 to 2009 under the leadership of Professor MRS Rao. Subsequently, we had 2 rounds of Center of Excellence on Molecular Neuro-oncology, that is between 2010 to 2015, 2016 to 2021, funded by DBT under my leadership. Professor Vani Santosh was a PI from NIMHANS in these programs and played a central role. I said earlier we have co-authored 45 papers. How can one judge the quality of these papers? This is important in today's world, which is facing mushrooming of journals. One can consider the impact factor of the journals where these papers are published and how often these are cited. Let us see where these 45 papers stand. Most papers are published in high impact factor journals and are also highly cited. Some journals worth mentioning are Oncogene, Cancer Research, Clinical Cancer Research, Modern Pathology, Journal of Proteome Research, Journal of Biological Research, PLOS ONE, Neuro-oncology, Scientific Reports, Molecular Cancer, Molecular Oncology, and OncoTarget. All the papers cited are cited at least 10 times, with 15 papers cited more than 50 times, 7 papers more than 100 times, 2 more than 200 times, and 1 more than 300 times. In addition to publishing papers in peer-reviewed journals, our work also resulted in the filing of 7 patents, 5 from CSIR and 2 from a DBT-funded project. While they are in different stages of acceptance, the US patent has been awarded for 3. Professor Vani Santosh is also an excellent teacher. While I am a trained molecular biologist, a little bit of my knowledge of neuropathology is learned from Professor Vani Santosh. Since I have 8 to 10 PhD students at any time working on different aspects of glioma, I used to bring the entire lab frequently to NIMHANS so that all my students could present their work and explain what they are doing. What is the molecule, their interest? Each of those meetings used to go for 2 to 3 hours. Every time I have seen Professor Vani Santosh patiently listen to them, ask questions, and get herself clarified on what exactly she needs to contribute to each of the projects. On a personal note, Professor Vani is a pleasant to talk and to interact with. The superannuation is only for the purpose of administration. I am sure her contribution to science, particularly neuropathology, will continue, and the scientific interaction between her and my laboratory will continue. I wish her a healthy and happy life for many more decades. Anybody else from the audience? Dr. Vijay.
So it was a very nice time when ma'am joined as a head of department after Sundar sir retired. So I joined in 2017, May 1st, 2017. So I knew most of very renowned scientists, and like I started working in oncology, hemat-onc in 2013 onwards. So the oncology field, before that it was new, but then slowly, and I was working in neighboring state, Hyderabad. So I knew Vani ma'am, Yasha ma'am, Anita ma'am by name. So it was dream to join here at NIMHANS, but no formal interaction initial few months. Maybe we met during faculty academic presentation. Then the Shara Puja and all that was but yeah, the one-to-one interaction I had first when I just walked into Vani ma'am chamber with my mother's slides. She was
Give some more.
Sorry. So I just went to ma'am chamber with few slides and tissue specimen of my mother, which was just 15 days before operated, and I wanted some opinion. So it was like assurance. I knew that I got information that it is going to be difficult, but still, ma'am assured. We had one-to-one talk. Ma'am explained fully that, okay, what we can do and what is going to happen next, what molecular thing we can do, and based on that, we can take that, what is the prognosis. But yeah, that happened. It's a destiny. So, okay, we were the new department. I joined after a few years, 1.5 years. Sundar sir retired, ma'am joined. So we had challenges at our department. Each department in the beginning will have their own challenges. So institute wise, and also ma'am guided us, like everyone who had interacted closely with ma'am knows that how like a mother guides with love, affection, the direction that she always know that where kids are going, what they should expect, what there should be the goal in the life. So she had set a path, and definitely we are trying to still begin our journey, but definitely, ma'am, I promise that we will achieve what you have set for us. So in coming years, the department, which is still, I say, in infancy, we are trying our best to put across, whether it is teaching or research. So it has helped us a lot. So I never, ma'am never said that whenever I call or I go to ma'am chamber that, no, I am busy. Despite I knew that ma'am is extremely busy, but many a time I just landed at ma'am chamber and ma'am is smiling, just put on Harsha, most of the time she will be discussing something with the ma'am and I'll end up there. And sorry, she has to wait for some time. Ma'am patiently listen, half an hour, 1 hour, whatever it is. So thank you so much, ma'am, for everything. Thank you.
I request my close friend, Dr. Naveen, old student of NIMHANS, to say a few words.
Good evening, everyone. I'm Dr. Naveen, old student of Vani ma'am. I was a student 12 years back. I'm still a student, of course. So I'm very fortunate to be madam's student. And of all her so many facets of her personality, she really stand apart is what I really appreciate is her patience. Because of her patience means she will never rush into a diagnosis. She'll take her time. Even for difficult cases, she'll tell, we keep on my table, I will see, give me some time. And next day she'll give a full detailed explanation of whatever the morphology is. So her emphasis on morphology is so much, she's inspired us that we should spend more and more time on the slides, which is so much important. Second aspect is her this thing, cool and calm attitude. I never seen her lose her temper. She'll always be smiling, and that has really helped us this thing over the years. And her third is the pursuit for excellence in all things. Because of that only, the department has gone to such heights, and such a beautiful lab has been done. And what few moments which I recollect is during my this thing postdoctoral fellowship is one when I made a right diagnosis. Right, means there was brain lesion, and the surgeon removed it, saying it was a tumor. But there was one small tiny fungus in one edge of the tissue, which everyone had missed, but I had picked up. So man was very happy and she really appreciated me for it. So I was very glad on that day. And one second moment which I really appreciate, which I recollect, is when I made the wrong diagnosis. That was outside NIMHANS when sir had some lesion in the neck, and it was they had come to HCG for second opinion, and then we had done FNAC. The cells looked really bad, and it was suspicious for some malignancy. But then again we had done a frozen, and it just turned out to be benign cyst with reactive atypia. We were so glad, and I could see the relief on man's face, and really. So all these words, congratulations, ma'am, for your superannuation, and all the best for your second innings. Thanks.
Thank you, Dr. Naveen. One of the main strength of Vani Madam is the student community and the student folk. So we have two sets of students, the PhD team and the DM fellowship student. Of course, not differentiating, just for the sake of compilation. So I request Dr. Kritika, was made a small compilation from one group, and she'll be presenting that.
Very good evening, everyone. Guru Brahma, Guru Vishnu, Guru Devo Maheshwaraha, Guru Sakshat Parabrahma, Tasmai Shri Gurave Namaha. The reason that I'm standing here today is because of my gurus, starting from my parents. And one of the gurus who has played a very big role in my life is Dr. Vani Santosh. I still remember the first day I met her. I was a project assistant in Dr. Kondeya's lab. Sir told me, go and meet Vani ma'am at NIMHANS. I called her. She said, she gave me an appointment. I went and met her. The warmth with which she spoke to me put me to ease, and I felt so comfortable around her. So that day I went home, I remember telling my parents, I really wish I get a position in her lab. She told me, you get a JRF, or you can join my lab if there's any project that comes up. And I think after 15 days after I met her, I cleared my CSIR and I went back to her. She had met me just once. Even then she was so happy, and she's like, I told this girl come with a fellowship, and now she's here with the fellowship. Now I have no option, I have to take her. I'm glad that day you welcomed me into your lab with open arms. I am standing here because of that, ma'am. So Dr. Vani Santosh, our beloved mentor, not just me, there are many students who have been trained in her lab who are doing really good in their careers today. And the times that I've spent in ma'am's lab, I can never forget. She had given me so much of freedom in the lab. I remember I was doing a lot of my work in IAC as well. I used to spend a lot of time in Kondya sir's lab as well. I was in touch with her always through emails. And if she doesn't get my email once in a week, she would text me, where are you? With the concern as to what has happened to the work, and also what has happened to this girl. Where is she? She's absconding. So when I come back, she would be like, welcome back. Where have you been? So, and the way every experiment, every small step of every experiment that I used to do, she was so curious to know everything about it. What did you do? Which cell line did you culture? Why did you take this cell line? What did you do with that cell line? How did you knock down? She had so many questions, and it was like it gave me so much of confidence when I was standing on any platform to present my work, that most of the questions had already come from Vani ma'am. No more questions left for the audience, and I was ready for any question that would come from the audience. She let me go to all the conferences, and since the time I joined, I was presenting at ISNO every year. All of my work was presented, and it gave me so much of confidence that now I'm confident to stand on any stage and speak. And I know what I have to speak, even though I don't have a script. I'm confident to stand on any stage and speak, and all that is because of you, ma'am. She was so particular about the way she trained us. I remember when I submitted my research proposal right in the beginning, the first draft that I took to her, I don't know what she felt. She did not tell me anything. She just told me, you send it to me by email, I'll let you know. From then on, I would always wait for her replies, which would come late in the night. After everything is done, she would sit late in the night, correct all the proposals or whatever we give her for corrections. And 12 o'clock, 12:30, I would have received her email for sure. If she says tonight I'm doing, tonight she will do it, come what may. And over a period of time, from my research proposal till my thesis, I would always see how less are the comments from Vani ma'am. That is like a promotion for me, that, okay, I've come closer to what she's expecting out of me. So I've learned a lot in this entire journey, ma'am. You continue to support me. Even I've submitted my thesis in 2019, and 1 year later, I went back to defend my thesis. And in that 1 year, a lot of things had happened in my personal life as well. 5 days before my defense, I went to her. She asked me, what were the drugs you treated your cell lines with? I was blank. I was totally blank. 5 days was my defense. She's like, don't worry, ma, go home, relax, go through your thesis and come back. So next day, I was confident to go back to her. So that comfort which she gives with every interaction that we have with her is what has, brought us all here. Ma'am, like you had a lot of fun with your colleagues in the department, we also had a lot of fun in the neuro-oncology lab. So we would go out for lunch, and all of our achievements, every student who is graduating from the lab was celebrated. And ma'am took all of us for lunch, and we had a lot of fun in the lab. So since we were a huge crowd, a lot of people were in Vani ma'am's lab, everybody wanted to talk about ma'am. When I contacted people to, share their testimonials, what they would like to say about ma'am, I have gotten some videos and some write-ups from a lot of your students, ma'am. Vani ma'am's first PhD student is Dr. Balram. So he has sent his apologies for not making a video and sending a write-up. I would like to just read what he has written. I would like to express my deepest sense of gratitude to Professor Vani Santosh for all the guidance, continuous support, and encouragement. Thank you, madam, for having trust and confidence in me as your first PhD student, and also for giving me a free rein in planning and executing my experiments, without which I would not have been able to complete the work in time, and for being extremely patient with me even during the most barren periods. I take this opportunity to ask for sorry for any small mistakes which I have done during my presence, and thanks for bearing with me so patiently. Thanks, madam, for the constructive inputs during my work, which enabled me to reach this important milestone in my life. This is from Balram. I just have a few videos of some of Vani ma'am's students. I'd like to quickly share them all. You can share it too in the meeting. I will also feel that you are busy. No, it's not.
One tea or coffee. It's coming here. Tea. Tea na ನೀವು tea ಇಲ್ಲ ಕಾಫಿ ನಾನು tea totally tea. Totally, it's not tea total. It's totally very It's so nice, Allah. So nice. I'm recalling what happened to me during my retirement. Nice. Huh? COVID batches. Yeah.
Okay, sorry, there's a technical glitch. We are not able to play the videos. We'll just try to sort it and come back and play it.
Okay, no problem. You can be
Henry.
We'll make you also sing one song as well.
We have another compilation by Dr. Shilpa, the first DM student and student of guided by Vani Madam.
Yeah, I'm representing the student community. I am a little bit emotional, but I'm extremely fortunate to be standing here as a student of a great teacher, Vani ma'am, who has definitely inspired all of us in many ways. And there are infinite qualities of ma'am, which makes you very special. But I will like to limit to 3 qualities which have influenced me to a great extent in the past 8 years that I've known you, ma'am. The first one is how, of course, everybody has spoken about your passion for neuro-oncology, and you have greatly influenced me in that I have developed an interest in neuro-oncology, and whatever little knowledge I have in neuro-oncology is all credit to you. But the love for diagnostics that you have, that is something very special, and I have actually learned to love diagnostics from you. More importantly is the way you approach your cases. It's not only for tumors, anything on the table, all the cases in the tray, anything, it's tumor, non-tumor, everything. The way you approach your cases is just inspiring. And whenever we have a difficult case, we run to you because the confidence that you instill in the diagnosis. If you say any diagnosis, you have no doubt in it. And that is a word you used. I have no doubt in the diagnosis, ma. And when you say that, I can confidently give my diagnosis, even if I'm not thinking about it. So that is something I have actually not seen in anybody. Whatever diagnosis you make it with absolute confidence. The second thing is, of course, how you have helped me, encouraged me at every step. You have helped me grow. You have given me a lot of opportunities. I entered this department as a PDF in 2014. I knew very little of neuropathology. I must admit that I knew very little of neuropathology. From then on, you started teaching, you're reporting, you give so much importance to all the minute details in your own words. If there is a small biopsy, you'll say, this is definitely pilocytic astrocytoma. And if I ask you, why, ma'am, why is it pilocytic astrocytoma? Just be very careful, meticulous. There'll be one eosinophilic granular body lurking around, and you just have to look for it, and you have your diagnosis. And then as a resident, you should be very careful when you're reporting with Vani ma'am. You have to search for mitosis. If you have not looked for mitosis, you're gone. And all these teachings have actually helped me. And now as a faculty, when I'm signing out the reports, I know the importance of all these teachings that you have provided. Then, of course, I started DM. I enrolled into DM DM neuropathology, and I did my thesis under you. That is when you took me into molecular neuro-oncology, and you made me feel like one among the team. You just took me in, and you have since then told me what to do, what not to do. The only other person that has told me like that is my mother. So you have handheld us till here. We'll definitely seek your advice in the future. The last virtue that I am influenced by, but I've not been able to take, till now I've tried, but I've not been able to inculcate, is how calm you are always. You're so composed. whatever comes, you're always smiling. Even if you have to tell us something not so nice, you'll tell it in the most gentlest way that is possible. We don't even realize that you have told us that we have gone wrong somewhere. You can just manage everything, so many responsibilities, but still you have not even once shown your stress on the others. It's just because of your commendable organization skills and your time management. You're just superb at it. Nobody can beat you on that. And I would like to end in your own way, in your style. You're an excellent mentor, ma'am. And on behalf of all the students, I must say that we are all blessed to be your student. We have learned a lot from you and we will continue to learn from you. We will seek your advice at every step. So I'm not going to say goodbye. You'll continue to be in our lives. So yeah, so I will now request Dr. Ankit to say a few words. Ankit, are you there?
Yeah, he's there.
Ma'am, can you hear me?
Yeah. Please go on.
Yeah. Good evening, ma'am. So it's a privilege for me to be given this opportunity to talk on such an occasion for our favorite Vani ma'am. There are a few qualities in her which I'd like to talk about. One is that how she has such a fantastic, unique way of promoting scientific temper in her students. I remember when I was working under her, in her own very charming way, she used to over appreciate all the good inputs her residents used to give. And then she also used to cheekily laugh away all the utterly stupid inputs we used to make on our cases. I still remember there was a case of germ cell tumor, and she was asking, what else can we do? And I came up with an answer, ma'am, we can do a pass stain, which was a relatively very stupid answer and very simplest way of doing things. And she was over appreciating me. She patted me on my back. Wow, wonderful. What an answer. So, she kind of gives you with a lot of confidence and she fills you with confidence. She also has a very cheeky, humorous side to her. And she understood the cockiness in my diagnostic aptitude. And I remember she used to, whenever I used to come out with some cocky diagnosis, she used to be like, Nodi, hudga tumba valle ide. So, some things I just can't forget about her. Honestly, I was just talking to Shilpa ma'am that I'm actually shocked that you're superannuating today. I always felt you're good to go for at least a decade. I really came as a shock for me. Ma'am, I thank you for all the learnings you have given me over the microscope and off it as well. I wish you all the good health ahead and a great life ahead. I would also like to end and finally, like to disclose and apologize I was the one who initiated that this party of yours on your birthday and we took all of you out and burnt a hole in your pocket. So, oh, I'm sorry for that, ma'am. I was the one. Thank you, ma'am.
Thank you, Ankit. Yeah, one thing that I missed saying is that ma'am loves food.
Yes.
And pizza is her favorite. So if anybody wants to,
Order something on sweet.
yeah, please order pizza and pasta for ma'am. So we'll move on. Dr. Ram.
Here it is. Is there?
Aniruddhan, are you there?
Move on to the next. Then come back.
Anusha?
Am I audible?
Yes.
Yeah. This is my, I am too emotional, though I'm not actually at NIMHANS with ma'am. I must say it was my privilege that I was one of her students. The first thing I remember is when I joined initially, the first few days, I was really, really scared of Vani ma'am. Don't ask me why, I was really, really scared of her. And the first mistake I did, some analysis of the glioma, I was really new to that new classification. And I was really scared that she's going to scold me. And she nicely told, it's not like that, sweetie. And she lovingly explained me the whole concept. And that is where I saw the loving side of her. And I was no more scared of her. And I think Shilpa ma'am has already told exactly how it is. You need to find the mitosis. And it's a must and mark it. I still remember her teaching that you.
Anusha, you got muted. Anusha?
Find those mitosis. So, that's really what I'm taught, and I still follow it. And one more thing is she really stressed on the morphology. I mean, though they were IFC, the way she used to find those subtle, softer findings, which will tell this has to be this. And ma'am used to tell, this will turn out to be this. And the best part is it would turn out to be that. So I think she made me fall in love with neuropathology daily there. And yes, I now proudly can say that I really want to pursue more of neuropathology only. And ma'am, thank you for always being there, always guiding. And I must say you were one stop for all solutions in neuropathology, and you still will be. Thank you for everything.
Thank you, Anusha. Dr. Aniruddhan, do you want to go?
There he is. There he is.
Yeah. Namaste and good evening to everyone. Let me begin by my Guru Vandana. Ajnata Thimirandasya Jnananjana Shalakaya Chakshur Unmilitam Yena Tasmai Shri Gurave Namaha. It's my salutations to my Guru who has lit the lamp of knowledge to me and removed all the darkness of Ajnana. Many people have told a lot of things about Vani ma'am. I'm sorry about the background noise. Many people have told a lot of things about Vani ma'am, and there is very little I can add. And especially one person was talking about the cycling skills of Vani ma'am. Perhaps he did not see the walking skills of Vani ma'am. It is indeed a pleasure to just see her walk through the corridor. That indeed, that it's a sight to behold, and that sort of a majesty doesn't come to all. That I would like to document. And I do not know. Madam was telling that nowadays students are more confident and more outspoken. I would really like to know who is that, who could do that to you. Is there any? I don't think anybody could have the courage to look up to you and talk back to you, because that is the amount of personality and radiance that you have. It is really impossible, and it is indeed a great pleasure to talk to her. And that sort of a personality comes not from external appearance, but from inner radiance. It is a sum total of her knowledge, her humility, and her absolute magnanimity of heart, and her confidence in herself. So all these things put together comes out as a radiance. And I would just like to share one one reporting incident with her. We were all sitting for the reporting, and Madam signed out a report saying that it was an astrocytoma grade 2 or grade 3. And she told that, Anirudh, I think this tumor will recur. And I was a bit skeptical at that time. So I was young and stupid. So I was a bit skeptical at that time. And lo and behold, 6 months later, the tumor recurred with a higher grade. And we were looking at the older case sheets. And at that time, Anita madam, with her inimitable wit, told that this is exactly what happens, Anirudh, when you contradict Vani madam in reporting. So that is her level of confidence of reporting. And people say that tumor do not read reports. But I have a nagging suspicion that the tumors you report actually read your report and behave exactly the same way as you have. So, I mean, I have the suspicion is always there. And the other thing is, one more thing which has to be told is that her extraordinary correlation with histopathology, her extraordinary grounding in histomorphology, whatever may be the molecular finding, she used to always say that you should always correlate with histopathology and find a feature which is reproducible. The molecular feature has to be reproducible in histopathology. So that is the greatest thing about Vani ma'am. Her, my only regret is that I did not get, I mean, I was unfortunate that I could not learn more from her. And my only regret is I could not live up to her standards and to her expectations. I'm really sorry for that. And actually today is a special day for me because today is my birthday. And thank you. But what better gift could I get than the Ashirvadams of all my gurus and especially Vani madam. Please bless me, madam, because that means a lot of things to me. And I would like to close by paraphrasing Shakespeare. Sorry, Anita madam, I'm going to paraphrase him, and I know you are laughing. I'm just paraphrasing Shakespeare from Julius Caesar. Her life was gentle, and the elements so mixed in her that nature might stand up and say to all the world, this was a dame. So thank you, the great dame of neuropathology.
Wow, wonderful.
I mean, that actually suits you, your personality. And I'm very sure, I have no doubt in my mind, to paraphrase Vani ma'am again, I have no doubt in my mind that she will excel in this phase of life as she has excelled in every other phase of her life. My humble pranams to you and all my gurus, madam. Thank you so much for giving me this opportunity. It's really wonderful to see all of you. Thank you so much. And I'm really sorry because about the background noise, I couldn't avoid it. I'm really sorry. Thank you so much.
Thank you, Anirudh. I think Nivedita wanted to be here, but she had to leave because it got late. Paramita has shared a message. We'll send it across to you, ma'am. Punkudi? Punkudi?
Yeah, ma'am. Can you hear me?
Yes.
Hi, ma'am. Good evening, everyone.
Good evening.
I'm very happy to share a few words about Vani ma'am on this special occasion of hers. There are just two things I would say. She, despite having a humongous knowledge and clarity in subject, she never looked down on anybody. Even to a first-year resident, she listens patiently, though we must be saying utterly something else as a description. She patiently listens, and then later she corrects us and guides us, and she tells us where to read and what to read. Yeah, we better read the next day because we will forget it, but she asks, so what did you read, ma? That's all. So we better read and come. And she's such a wonderful teacher. And ma'am, this world needs more people like you. I always wondered, ma'am, about your ability to multitask, how she does it. I always think she has a huge commitment list, regular reporting to research work, to administration work, but still she looks always calm and composed and always bubbly and fresh. How she manages that, I really don't know. I always wanted to ask her the secret of that, but I never dare to. She's definitely a superwoman that way. She's beautiful in and out, and she has a great sense of humor, of course, admixed with the subtle sarcasms. That sarcasms which I most times miss. The very good ma has actually meant, ma, you need to improve, which I most times miss. Yeah, but I'm thankful to you, ma'am. Thank you for being an inspiration to all of us. I'm sure you will have a blessed second innings with a full Josh as always. And thank you, everyone. Thank you, organizers.
On behalf of all residents here, I would like to say a few words. And whenever anyone has to say anything about you, Vani ma'am, the first thing which comes to our mind is obviously that you're a great teacher and a great mentor. You force us to think about any case we are seeing. After we have read about the case, we have described our findings, you will always ask, so now what do you think? What will you do next about this case? That forces us to think much more into the case than just giving a diagnosis. And you are very strict. Ma'am will always want a precise answer to everything. If we try to go about in a roundabout manner, she will say, I've asked you a specific question and I want a specific answer to it. But at the same time, you're a great motivator. For every little answer we give, you will say, wonderful, excellent, ma. And it really brings a smile to our face. She's a great enthusiast. She's not only enthusiastic about neuro-oncology, but any discussion on nerve and muscle pathology we've had, any discussion on the new hospital information system, she's upright and giving her inputs in everything. Any birthday parties, she's the youngest resident there. She's our walking encyclopedia and also are ready to refer full text version of all the 3,624 references in the new WHO CNS classification. We all love you a lot, ma'am, and we all thank you for all your teachings. We hope that we don't miss you much. We hope that you keep visiting the department because each time you visit, we'll heartily welcome you with a whole lot of praise of unresolved cases which need your opinion. Thank you so much, ma'am.
Thank you.
Dr. Ishani, are you there?
Yeah, I'm here.
Please go ahead.
Can you hear? Sorry.
Yes, please.
To start with, I'm very thankful and grateful to Anita ma'am and Dr. Shilpa for giving opportunity to be a part of this symposium. It's an honor to be honored, and I was very fortunate enough to be with Vani ma'am as her student. I really don't have words enough to describe my relationship and my feelings for her. She not only was a teacher to me, she taught me to become a helpful, optimist, humble, lovable, ethical, and inclusive person in workplace and anywhere that I go. These are the few attributes that she has, apart from being the most calm and composed person I've ever seen in difficult times. She was always just a phone call away, and her voice on the other end, hi Ishani, would really make my day. Thank you, ma'am, for mentoring me. And I must say, she is the one who is instrumental in starting my journey as a consultant in neuropathology at Medanta. Thank you, ma'am, so much. And I am really, really fortunate that I was your student. Those were my best days there. That 1 year, I really cannot forget what I have learned there. Wishing you all the best and also good health in times to come. Thank you, ma'am.
Thank you, Ishani.
I'll now request Dr. Vidya to speak.
Congratulations, ma'am, for touching the key milestone of your career. Actually, everybody has already spoken so much, and I have so many things to talk, but I'm really not getting the words what to talk. Actually, today I'm standing here because of sir. So first of all, I should thank sir for introducing me to madam. So I don't know, like all of you know, sir is pediatrician for my kid. And it was first time in 2013 or 14, when I had gone to sir, he was talking to me and he asked me about my, what you do. I told I'm into research. That time I was working in St. John's Research Institute. And then sir told, okay, my wife, she works in NIMHANS and all that. And then he called ma'am down and introduced me that she's our family friend and all. And then madam gave me her card. And then ma'am told, okay, in future, if you want to work or if you need any help, you can contact me. And it so happened that after 1 year, my PhD registration couldn't happen in St. John's Research Institute. And then I was looking for other opportunities. And that time I remembered madam. And I went to madam. And when I called madam, that time, first time, she was traveling to Mumbai for some conference. And she's like, Ma, I'm not there in NIMHANS now. Today you don't come, you come some other time. And then slowly, the project on which Kritika was working, the project kind of was set for me. So it was easy for me to take over. And I had a limited time because of my fellowship. So it was like perfect fit for me. And throughout my journey, I can't tell you the way madam has helped me. The madam's discipline and the way madam teaches us everything is to think about the work, experiment, everything. You just can't do it. You have to think why you did that. We have to explain her why we did that. I remember, especially when we were setting this molecular oncology test, especially qPCR assay for MGMT, third promoter mutation, and all those things. That time, we used to discuss among ourselves, and then we'll tell this to ma'am. But telling to ma'am and convincing her, ma'am, we want to do this, we want to explore this kit is good, that kit is good. Then ma'am, as everybody said, no, madam will listen first, and she will not say anything. And once she's convinced, then madam will say, okay, you tell me now what you want to do, what all you need. And the kind of freedom madam gave us to plan our experiment, to take these things ahead, it's really, really helping me in my current job now. Not only this, how to present scientific work, how to write it. As Kritika said, I still remember my first manuscript when I had written, it was full red. And ma'am, what you wrote, ma? It should be like this. Then second draft, it was little better. And then I went on modifying my drafts and it became better. And to tell you, the reviewer's comment was the best, very good written paper actually. That was one of the comments they had, very well written paper. That was the reviewer's comment actually. And it was not like, madam, she will write for us or something. She will make us write and she will guide us how to do it, how to write. So the way madam has trained us, it's really helping us now outside, you know. And I should say it's like a small kid which like who had just started walking. Like we passed out, we finished our PhD, we have gone out and we are still in that, you know, the kid who learns to walk and still will be looking behind whether somebody is supporting me or not, like that. Madam is like that, in that position. Every time she'll ask, how is your work, ma? Are you happy with your job? What are you doing? All those things. And always she'll be supporting her. always do your best, all those things. So as everybody said that ma'am's energy level and the confidence, that is really a source of inspiration for all of us, ma'am. So we are so proud of you, and like we are so blessed to have a mentor teacher like you. Thank you, ma'am. And I wish you a happy journey ahead and a happy, healthy, joyful life with your family. Sautri? You can talk. You are muted. We'll ask Harsha to speak and then we can talk.
Hello, can you hear me, ma'am?
Yes, we can hear you.
Okay. So congratulations, Vani ma'am, on your superannuation. So I'm not a great speaker and lot to speak right now. I'm getting emotional. So I'll try to keep it brief. Ma'am, I was fortunate enough to have you as my teacher, guide, advisor, and mentor. I have to confess that throughout the first few months of my residency, I was just amazed to see how devoted you were, and as they said, your energy level and how passionate you were towards the neuropathology as a whole, not only neuro-oncology. So we all know about your contribution towards the subject. What else to say? But what is Madam's influence in the lives of our students? First of all, the constant motivation and inspiration to do some good productive work. Madam always used to say, always urged us to carry on good work. And she has been always very inspirational and helpful towards students, especially from the very beginning, which have helped us help to build up the immense confidence level in the freshers, in the students. And technically speaking, as a pathologist, I learned how to frame a histopathology report properly, and the proper approach to diagnosis, to diagnose any case, however difficult it is, the proper approach to a diagnosis. And she is one of the most sensitive and respectable person I have ever met. She was strict, very strict when needed, at the same time very humble. Thank you, ma'am, for all the memories and for the most precious years of my life. You will always be with us, and you keep on guiding us. Stay healthy, ma'am, and stay happy.
Thank you, Sautri. Good evening, all. And I'd like to thank Department of Neuropathology for giving me this wonderful opportunity to talk few words about my mentor, my teacher. It's been like 10 years since I had joined Neuropathology department. And madam says that I'm still a baby PhD student, but I have stayed with madam since 10 years, and she has nurtured me in all manners. And I still remember when I came for the first time with my mom, and she greeted me like her child. She took me into her warmth, and how she what do you say? She I was like a stone, and she was a sculpture who hit me and made a proper. Still not at that part, but still I'm improving a lot. And a few things that
we all PhD students are from a basic background, and we usually look at the moleculars of the tumor. But madam, she would tell us, no, don't only stress on moleculars. Study moleculars in light of pathology. That will help the diagnosis of the patient. That really, when whenever we are at the lab and we get cases, then we'll see the reports what have come, and we'll say, no, madam, we can do this. Then madam will be like happy with us also that yeah, this girl is giving a proper suggestions. So, and I cherish those fish when we usually madam sits for fish with me, and she'll tell you have to be there. Then I'll be like very proud that I'll be sitting with madam and seeing the slides and all. So those are the memorable moments for me. And even teaching wise, she would give her teaching slides to us, though we are not a pathologist, but she was giving us her teaching slides, and she would tell at least attempt for the differential diagnosis, and that helped us a lot. Sure, Vidya will agree on this. She would give us various tumor related slides, and just give a differential diagnosis. Then we would go back, read, and come back to madam and say, yes ma'am, we also have read it. Then that was like a enthusiasm which madam gave us to look at the various other tumors. And one more thing is, she's actually like a mother for me, because recently I holded my daughter's birthday, and she came there, and it was so fun. It was like a family member. She came there, and she enjoyed with us. So thank you, ma'am, for that. And and additionally, I want to tell that our neuro-oncology workshop was successful because madam interacted with each and every participants there, and she stayed back in the evenings, saw their slides, and gave necessary suggestions. Because of that, it was successful. Ma'am, I would like to wish you a great second innings and a healthy second innings, ma'am. Thank you so much.
I'm reading out a message from Dr. Aparna. NIMHANS is going to miss a great teacher. I heard ma'am first at Tirunelveli way back in 2001 during my postgraduate days, and the crisp way of her presentation put me into the long list of admirers of this young and vibrant teacher. My tenure as a postdoctoral fellow at Neuropathology NIMHANS helped me know Vani ma'am more, and I had the good fortune to do my project and publications under her. She is an ardent researcher, very particular about her scientific work, and allowed no inertia as far as projects were concerned. The lighter moments spent in the department brought out her motherly affection. Ma'am is ever ready to answer queries and offer her opinion on the umpteen number of cases I sent her. She kindly obliged to be faculty at our postgraduate teaching programs many a time. Although she is superannuating, her immense knowledge will be a guiding light to the coming generation of neuroscientists. Wishing ma'am and family good health, happiness, and peace in the coming years with a lot of love and respect.
Thank you, everybody. Last and most important is the family. We have a message from Nikhil. One second, I'll just. One second, I will just. Share. This one. Yeah.
Can't hear. Doing so fast.
ಇಲ್ಲಿಂದ ಜಾಯ್ನ್ ಆಗ್ಬೇಕು ಆಡಿಯೋ ಮತ್ತೆ ಆಡಿಯೋ ನೀವೇ ಮಾಡ್ಬೇಕು. ಆಡಿಯೋ ಇಲ್ಲಿಂದ ಜಾಯ್ನ್ ಆಗ್ಬೇಕು. ಇದೆ ಶೇರ್ ಸೌಂಡ್ ಇದೆ.
At least we can hear them. I'm lost for words, really. I don't have any words to. I mean,
Thank you so much for organizing this wonderful send-off event for my mother. I'm really grateful that she has such wonderful colleagues and the friends that she has made over the years at NIMHANS. I wanted to share a little bit about how much of an influence my mother has been with her work and through her career. One of my earliest memories is this one time that my mom left home to go to work. And I remember asking my grandfather, my father's dad, what she did at NIMHANS. And I remember him proudly saying that she solved puzzles of the brain. This is probably why I used to think that NIMHANS stood for National Institute of Mental People. My dad loves to make that joke. I did not fully understand the impact of her work until I was about 12 years old. It was the first international conference that she had been invited to. It was in Bali, Indonesia, and she brought me along. I attended her oral presentation of some work she was doing on tumor markers or some such, and I was just amazed by her clarity of thought and the presentation. And had not seen anything like this at school. It was just a whole nother league from my school teachers. And there were these other experts in the audience who stood up and asked what seemed like really challenging questions. And she just handled it with great aplomb. And this was the first time I was exposed to research and real knowledge dissemination. And I loved every bit of it. Over the years, I've helped my mother out with her PowerPoint presentations for various conferences, and I've just learned a lot about how to structure a presentation in a manner that captivates the audience. It influenced me tremendously. And in fact, that was one of the reasons I chose a path of research myself, and I pursued a master's and a PhD with an ultimate goal of becoming an academic. Although my career took a different path as I was drawn by capitalism and decided to become an entrepreneur, but that's a whole nother story. My mom has been a beacon of light for me through the years. I'm just so proud of her accomplishments that we're celebrating here today. While this is the end of her first innings, I can't wait to see what she does in her second innings. I love you, Mom, and I truly wish I could have been there in person to celebrate with you.
Should we end the program or what?
To sing a song.
Yeah, because everyone is like still enjoying, continuing, listening. So don't feel like going home. Thank you each and everyone for an active participation. And I don't know what to say at the end. I just want to make a very warm wish and prayers for Madam's health, prosperity, peace and happiness in the next phase of life. And also to bless us and to support and guide us in every aspect, and good health to sir and everyone. And thank you everyone.
There are very few people, so it's okay. I can just talk like that only without any mic or anything like that. But it's truly amazing to listen to so many colleagues and students and what you feel about me. I didn't think that I would get such good words, really. I don't know whether I deserve all that, but it makes me feel so good. And my stay at NIMHANS, it makes me feel like I've completed the, I mean, what I was expected or what I had to do. I think I have done that. And along the line, if I've hurt anybody, if I have said anything that you didn't like, you please forgive me for that. But at the same time, I think students will help us to grow, and they help us to. Every time I used to feel very stressed about something and I'm with a group of students, I forget everything. And it's just that students and, they share so many things with you, and it's always such a lot of laughter, such a lot of fun. But at the same time, we are learning so much. We are learning together. We are continuing to learn, of course. And with great friends like Ari, I have Vijay, I have Param, everybody who is staying till the end of the program who are here. And thank you all so much. And yeah, you're going to be seeing me. So it's not that I'm going to disappear or something like that. Santosh knows that. And he keeps saying, for the next 3-4 months, I won't even bother you because you won't be at home. I know that you'll have something to do. He keeps saying. So anyway, it is a second innings that I have to start. I don't know where I'll start, how I'll start. I am not very sure about that. But I think I will take a little break. I just want to spend some time with my husband. I just want to spend some time with Nikhil and his family. And I think I need that break a little bit. Then I'll jump back. I'll come back and I'll bother all of you once again. So that is definitely there. And I wish everybody the very best. I wish the department the very best. And thank Anita so much of interest. Anita, Yasha, Nandish, Shilpa, Rashmi, everyone have taken so much of interest to organize this symposium, which I thoroughly enjoyed. Whatever you all said, I don't know, but I enjoyed every lecture starting from the word go with Ari till the end, the last speaker. I was very happy, and I'm so glad that Santosh is with me today. He is always busy with his own patients, his own things, but today he said he'll spend some quality time with me, so I'm very happy for that. And yeah, that's it. Thank you all, and I love you all.